Awesome. Looks like we're live here tonight on Standing for Truth. We got some awesome guests here, all highly intellectual individuals. I'm choking on the food I'm eating. Jeremy, John, Matt, we're going to be talking about all things genetic entropy. So we're going to be talking about genetic entropy not only in humans, but in the fossil record, in animals, bacteria, viruses. I mean, the fact of genetic entropy is inescapable, but the evolutionists are always running. They're running as fast as they can, but they can't get away from the fact of genetic entropy. So you have to get some water here. But that being said, Matt, you're going to be starting it off with animals, right?
That's right. Well, I'm going to talk about everything but humans this time. I'm briefly going to touch on some plants, and then I'm going to talk to you about Haldane's dilemma and why that's a problem for evolution. Then I'm going to give you some examples of why it's a problem, and then I'm going to talk to you about a couple genes, and then I'm going to give you guys some visuals, and I'm going to give you some examples of how genetic entropy is occurring in animals. So I will just jump into this real quick by talking about cassava. Now, that's a tropical plant that you guys know as tapioca. They've recently done a study in 2017, and they found when they tested the plants worldwide that the cassava plant is riddled and has accumulated many deleterious mutations. It's showing clear genetic load and a clear fitness decline as well. And remember what Haldane said. He said, if we see one mutation per generation, we are in trouble. And now we know that that number is around 63 to 100 every generation. And that became one of his conundrums. But here's another thing. He also said that we can allow about 1,000 beneficial mutations to become fixed within the pre-existing human population of about 6,000 years. Okay, so this is the big problem now. How many mutations have become fixed in humans and chimpanzees? Well, it's about 30 million have become fixed. So that's Haldane's dilemma. So now you can see the accumulation of deleterious mutations. It's just a huge problem. We also have deletions. We have independent gene loss, and the independent gene loss in multiple lineages. For example, flies, worms, yeast, and plants. Now, the reason that I'm going to bring up flies and worms and yeast is because we test on those a lot as human beings, because genetically we're very similar in a lot of regards to these animals and creatures. So they have such a fast generational time that we do most of our experiments on them. It makes no sense to experiment so much on other wildlife because of the fact that they don't have a quick generational time. It's hard to study and it's expensive. So we also have, no horizontal gene transfer from bacteria. There's broken genes. We have broken genes, we have copying errors, we have replication errors, we have gene loss, we have copper number variances, we have basically degradation over time. We have frame shift mutations that weaken things. And most of all, we have one particular gene that's in all animal life too as well. It's a broken gene called MTHFR. And this gene regulates how well others' genes work, and especially through methylation. So that even inhibits our epigenetic regulation. So the more broken that gene is, the more broken other genes are. And all animal life has a messed up version of this gene in some way, shape, or form. So now I thought it would be really fun to give a visual representation of what's going on. So I'm going to share my screen for a second and give you a rundown of what I see. And I'm going to bounce over here. And what you're looking at the top right here is Noah's Ark. And so what we're going to do is we're going to look at chromosome count. And we know what recent species have evolved recently. We can see the red fox down here. We know when that one's been kind of, when that arose. So we know what recent animals are, but we've never seen, obviously, the bat-eared fox evolve out of anything. So when we go back, there's a couple ways to determine what came off of an ark. I like the CO1 gene. That's one of my favorites. We can also look at how much heterozygosity they have. So we'll get into that. But if we look at the chromosome count right off the ark, we can see a clear declining rate going all the way down into current new species. And we can look at the heterozygosity right here, and we can look at the modern red fox, which is right here. They have some of the lowest rates. And then we look at the original, what came right off the ark, and we find the highest levels inside of them right here. So quite interesting, right? Now, yes, of course, this can be reversed based on if this bat-eared fox decided it was going to migrate and it got trapped on a small island. Sure, there would be that case, but you'll see that this scenario that you're looking at through speciation is down the line. Now, what I could do real quick is actually, this just occurred to me. I'm going to give you another visual representation before I keep scrolling down and showing you more of this. I'm going to go up to genetic drift as an example. And look at that. That's all over the place. I use that a lot in here. Here we go. This is a really good one. Here I put some animals on the top of the pyramid. Now, speciation says that animals started down here at the base of the pyramid, and they go up from mutations. And sorry, I just read something funny on my text. Anyway, the animals from evolution are going up. We say no, they're going declining. We're saying that these creatures came off the ark, and through genetic drift, they have been speciating and going down. And we're seeing a loss of information as they go down. We see a cornering of a species. We see higher amounts of heterozygosity from these species, and as they go through time, they're going down. They're boxing themselves off and becoming more homozygous, and we're seeing mutational loads accumulating and mutation rates. So this is a visual representation. So what they're saying is that animals in evolution are trying to go up this, and they're going up it on all different sides of the pyramid. And all of a sudden, when there's a problem, they said that they can jump up this thing very, very high. So they're making extreme leaps, far more extreme assumptions than we would make in our model, and impossible leaps, actually, considering we say that those scenarios don't happen. So they're actually invoking far more magical scenarios than we ever would. And I'm trying to go back here to my other visuals for you. I don't know where I put it. Oh, here we go. So this is the first one. I studied foxes real quick for you guys. So here's another one. Here's the horse kind. So we know the wild horses are these. They have the highest amount of chromosomes. They're the 66. They're the last wild breed. A matter of fact, these horses are untamable. They used them in the war, and they put them in zoos, and they stopped mating because of it. They almost became extinct. They were down to like, I think, 12 horses at one point in history. So they would not do anything. They had to release them back into the wild just so they survived. And then we have the normal horse, dropped to 64 chromosomes. Then the mule, which was a hybridization, you know, and then we got 63, and then the donkey 62. And then we have the last species to evolve, which was the mountain zebra, down to 33. So we see clear trajectory here, everybody. We're seeing loss of chromosomes over time. And this is the first evidence. I'm going to give you 7 different proofs of this. Now we go to the pig kind, or the swine. We have the wild boar. Now, this was, again, an original right here. We're looking at 38 chromosomes, and over time, boom, loss, speciation occurs. They start shifting from their natural state again. We have a higher observational rate of heterozygosity in the original wild one, and we see the loss. And when we get down here to the giant forest hog, which is down to 32 chromosomes. And you can see during the study that they've diversified. They're everywhere. You can find them all over the place, but yet they have the highest amount of homozygosity. And that usually is only the case, again, through the evolutionary model, if they're stuck in an evolutionary bottleneck, or they're isolated, or they're inbred. But yet we don't find this as the case with a lot of these animals, for example, the zebras and these red foxes, but yet they are the most homozygous. So then again, Dr. Jensen has been vindicated yet again with making his predictions, and showing his model is very superior. Then we go to the buffalo kind right off the ark. We have the African buffalo. That's what they named it, because it's littered all throughout Africa now. It has the highest amount of chromosome count, 56. And then we go down to the swamp buffalo and the river buffalo, the more common versions, and the American bison. And as you can see, the clear trajectory again, loss, going down. And again, I show the amount of heterozygosity plummeting down into that. And then I ran out of time making these charts for everybody. So I'm going to talk a little bit about mutation rates. I actually show some right here. Here is the last tested big versions of humans and chimpanzees. They averaged about 0.6 mutations per generation, so that's less than 1. Again, we find gorillas are a little bit higher. Right here, they have 0.82. So we can see the correlation. Again, the mutation rate is very quick. It's higher in gorillas than it is for humans and chimpanzees, but there's a reason for that. And again, it's also higher in, matter of fact, here, read this. Here's the Lowland Gorillas. They're going through a clear state of genetic meltdown. And what's ironic about this is they're not interbreeding, but yet we have an alarming rate of mutations that have crept into their genome, and they can't get rid of them now. They basically cornered themselves genetically. So they're in huge trouble. It's just a matter of time of when they go extinct now. And here's another one, the wild chicken, the Gallus gallus. Now, of course, we have regular chickens. That's what most people have on their farm. But they wanted to go and test wild chickens. So you can find this study right here. Of course, they didn't use a picture of a wild chicken because it's just a news article. So they don't know the difference between them. But you can find that they actually tested their mitochondrial mutation rate. Usually that's rare. You're going to find the nuclear mutation rate. They actually found it 15 times higher than phylogeny. So again, they disproved themselves on evolution. And we find that their mutation rate, look at that, very similar to human beings, right there, right on the cusp. And that was not what they expected, as usual, again. So here's another one. Is it evolution, which has no genetic, which is the no genetic impact hypothesis, or is it Jensen's, the created heterozygosity model? So I would like people to, take your time and read through this later. You will clearly see which model is more understandable when you're going to be looking at these different things. Not to mention that we have clear mutation rates. Look at this right here. The mutation rates between species directly correlate with what's going to come off the arc, but we're going to stick to genetic entropy at this moment. That doesn't deal exactly with speciation entirely. We're going to be talking about a little bit of deletions and why that matters. And I would also like to talk again about mutation rates, because that is very, very clear. And we directly observe it in wild animals. No longer can they say that, oh, these are just humans that you guys are comparing us to. No, absolutely not. We know everything there is to know about worms, for example. We study worms and fruit flies more than anything. We've mapped the entire genome of worms. And the reason why is because we share about 50 to 60% of our genome with worms. So the more we understand about their genome and sickness and disease, even with horses, we've actually found that we have about 90 different diseases that are correlated with humans that are in human beings. So we've mapped their genome as well. And every time we correlate and look at their mutation rate, we find the same problem. Matter of fact, this study right here was just linked to me by Standing for Truth. And here's the mutation rates that show that the correlation between all mammals is identical with one another. So I haven't got a chance to read through it yet, but I can link that for everybody on the side. This is going to be a really good one for me to read through later. And here's the study that I linked for everybody earlier. So here's the name of that if you want to read through the genomic meltdown occurring in the primates. Here's another good study that I found looking at bears. They're going through they're saying that they are resistant to genomic meltdown. That's a really fun study to figure out why they said such a thing. And I find that ironic. And here's another one. This was a shocking one for him. We'll read through that in just a moment. That was actually what I linked earlier here. And it's very fascinating, because what you'll actually see is they said that no species are driven to extinction before genetic factors impact them, mess with them. So here you're going to find that they actually tested a bunch of different species, and they went down and they looked at their heterozygosity levels to make sure that they were testing, good wild animals. And they actually found that genetic entropy was a clear thing occurring in wild animals. And most importantly, non-threatened animals, meaning they're not in some zoo and they're not bottlenecked, meaning they're not inbred and stuck on some island somewhere. So that's the most important thing, because a lot of people says, well, of course the mammoths died from high deleterious mutation rates because they were inbred and they were bottlenecked and they were cornered genetically. This counters that argument, because here they test multiple different things. And I dare anybody to go out and find anything that you guys desire and get a mutation rate. And that's what Jensen has done. Dr. Jensen has clearly stated, we can go into the wild. You can pick any animal that you desire, bring it back to the lab. You make your predictions based on phylogeny and evolution. I'll make my predictions based on observable rates that we see and the creation model, and we'll see who wins. Obviously, nobody's taken them up on that, because they realize and they know that the constant rate of deleterious mutations that are building up in the body, even near neutral ones, is more than the genome should actually have and can bear. And the clear trajectory isn't good for them, because even if a bottleneck occurs, well, guess what? If I've accumulated in myself more mutations in my family, and I've accumulated, let's say, 18,000 mutations, and I go into a bottleneck, well, guess what? I'm still going to pass that on to my offspring coming in from that bottleneck. So they're just going to inherit every problem that I had and pass that on to the new world. And now they're limited to whatever genetic discrepancies I might have had in my body. So again, that goes back to animals. By looking at genetic drift, we can clearly see that. HIV, here's another one. Here's another thing that's not a human. HIV sequences, they've been obtained. They go back right what we would expect. HIV is a great observational mutation rate that shows it's young. I'm glad that popped up right there. Didn't even expect that. There's all kinds of things. Skin color, that's another good one. But then again, that goes back to humans. So I'm really trying to stay on focus here, guys. Here's a good one. We can look at the allele shifts going on. We know that when there's a bunch of capital letters or when there's a bunch of lowercase letters, you have dominant, recessive, and then they can be swapped. So there's a 50% chance from both parents as the animal that had stripes would actually have stripes as well. So again, we're seeing that through time that the alleles are being shifted into a point where they're bottlenecked and they no longer can pull themselves out. For example, if you have a horse that has all dark fur and it's dominant and it bottlenecked itself, it would only be able to produce a horse that doesn't have black fur if it mated with another horse that would say white. So therefore it has that opportunity. But that's what we're seeing in nature when we look around. We see this clear dominant and recessive information being whittled down into a successive loss over time from this shifting of heterozygous gene sites to homozygous. And what the actual play is not evolutionary, what they're telling us, meaning it's genetic drift, not natural selection. They invoke natural selection for everything based on beneficial mutations. We say that the model, what actually makes the model functional is genetic drift. And then when we look at point mutations, that's a problem because most of the time point mutations are gone from deletions. So they're losing information again. So we see genetic drifts, shifts, loss, and that's what we're talking about here. So I think that could wrap it up unless you want me to go on, but I know I'm kind of running out of time. Unless you guys want me to specify anything, let me know.
No, I think the audience should definitely check out that paper that you screen shared there that shows mutations in mammals are all essentially the same. Because Matt, as you were pointing out, for example, in the mitochondrial DNA clock, and even in the Y chromosome, they're essentially fast in all mammals. Isn't that right?
Yes, exactly. And what you were looking at was just what that study is mammals. So we're looking at mice and primates and humans.
Right.
What I was showing is I wanted to go to horses. I wanted to show people that it happens in wild chickens, because
Right.
like I said, they're not studying everything. So it's really hard to find these studies, especially when it comes to mitochondrial DNA rates. So when I find one, I immediately store it for us and let our audience see it. And every time the scenario is exactly the same and the evidence is the same, it's very clear that the mutation rates are identical and they're fast. They always invalidate phylogeny, and phylogeny is the evolutionary assumption rates. And trio methods or pedigree methods are the creationist model. That's what we see. That's why they point to a young Earth, a young mitochondrial Eve, and the evolutionists can't deal with that fact.
Right.
So they try to say, oh, it's different in animals, and now you guys are seeing it's not different.
Well, remember, they have to invoke slower mutation rates in the past that are like 10 to 20-fold slower than is actually observed today. Yeah, so if it fits with our model. And you showed, I posted the paper in the chat, but you showed that example from the tropical crop cassava that they're accumulating many deleterious mutations. Right. So there's a huge decline in fitness even in plants as well.
Right, exactly. No matter where we look, so they're kind of in trouble. You know what I mean? They're in deep trouble. That's why they invoke their own methods and their own models, because things don't match what they need to match. So they just come up with a new model, and then they invent a theory, and then they invent a model, and then they say, look, it proves it. It lines up. Well, yeah, you just invented it. Of course it works. It's like if I invent a new shoelace because my shoelaces are broken, does that prove that I invented, like, oh man, I invented shoes? No, it doesn't. It's just
it's ironic that they love to use the puddle analogy again and say that that's what we do in context of our models. When I would say phylogeny and different things you're talking about would showcase a quite literal exemplification of drawing the bullseye around the target or the bullet, I guess, once it's already landed.
You're exactly right. Yeah, I mean, like I was saying at the beginning, they pretty much have to run from this evidence because even in really good presentation, Matt, I like how you showcase the shifts from heterozygosity to homozygosity from not only creation, but from the flood. Just like these horse species, how they have more homozygous gene sites now. Because, as you pointed out, we're looking back in time, according to our model, 6,000 years ago at creation and 4,500 years ago to the flood. We're looking back to the expansion of the genome. There's been a reduction of functional information since the archetypes started moving and speciating, for example. While the evolutionists, they look back in time to the contraction of the genome, right? They would need to explain the expansion of the genome to explain their single-celled organism to whales and just explaining all the biodiversity of life. So it's just fascinating that you're right. Yeah, for the audience sake, it's because a lot of the times, and Matt, we were talking about this earlier, the evolutionists will concede that obviously man is degenerating. They were clearly accumulating so many mutations to the point that natural selection can't eliminate, especially the nearly neutral ones. And as you always point out, I mean, we're seeing thousands and thousands of new genetic related diseases every single year. So there's very few evolutionists that would actually admit that man is getting better. Clearly man is devolving, but what do they resort it to? Or what do they explain it away as, Matt?
Well, they invoke that, as far as mutation rates, you mean? What are they?
Well, they'll agree, okay, yeah, man is evolving, but it's because of the lack of natural selection.
Oh,
right?
Yeah, selective pressure.
Right. We've managed our own environment, for example. We take care of the sick. Somebody is born with a highly detrimental mutation, we'll take care of that person, right? But in the wild, natural selection eliminates the weaker, right? Natural selection will eliminate that zebra that's born with a defect that can't run as fast from the lion, for example, So they'll just say, yeah, but in the wild, there's no genomic degeneration. And yet, as you've proven here, in all forms of life, plants, mammals, we see it in viruses, we see it in bacteria, for example, in Lenski's experiment. So,
well, and I think the irony of the part of the point you made there, man, was according to their model, somehow that zebra that started to sprout a wing would somehow survive the lion.
Right. Or somehow, if that zebra went into a bottleneck, we would get all the other horses on the world today. And that's clearly not the case, because that zebra has actually lost so much genetic information and shifted to this homozygosity state that the allele shift opportunities to have more offspring and speciate is hugely limited. So, for example, the Mouflon sheep, or what we say came off the ark, they have the highest amount of chromosomes. They have a huge genome. They have, we would say that they came off of the ark. And so they started speciating, and we have modern day dilapidated sheep that really don't do much except for that we shave down for fur. So what happens, or wool, I'm sorry. So what happens is when they get left on an island, they actually don't have a problem, if you notice that, because they are the highest amount of heterozygosity animal in the world. So when this actually happened on the Crumbling Islands, they actually speciated and lived just fine. No problem at all, because that's what we would expect. And it was completely contrary to what the evolutionary prediction was, in the fact that if you were to take a modern day sheep and put those on the islands, they would have a huge problem because of inbreeding, because they have already made that shift. So they need more genetic diversity arising in animals, and they're doing the exact opposite. They're seeing a reduction in genetic diversity over time. That's our model.
Well, as a quick interjection, something I was thinking about as you were discussing the greater numbers of chromosomes in these ones that you're defining as, the ones that came off the ark. When you think about that in the context of, what was the purpose of all of these extra chromosomes, right? And to me, what's really striking me is, okay, that is the pre-programmed information requirements for the different types of species that are going to come from that origin kind, if you will. But then why, when you think about it, why do humans have so few? And one of the things that was really striking me as you were talking about that was, well, yes, we have diversity, very, very limited diversity in our genome, right? But there are literally no other species of humans. So to me, it strikes me that God created the ability for us to have, yes, humans as our own species, to have diversity within our species, but not additional ones. And because I thought about that quite a bit in context of some of these different, worms that have a bazillion chromosomes, and so the bulls you were talking, or the African bulls you were talking about, like, what, 68, 64, something like that. I mean, that's almost 3 times as much as we have, right? And when you think about that, I'm like, we weren't pre-programmed they were pre-programmed for speciation. We were not.
Right. Yeah,
however, in reality, we actually have a much more complicated genome, even though it is smaller from a chromosomal perspective.
Right. Yeah, no, that's a fascinating point you make there, John, because it does look like, based on our low genetic diversity, low chromosome count, that God did create us, humans. Well, and directly from the Bible, it says that he created only 2 humans, Adam and Eve. So that makes sense that's what we observe is low genetic diversity. It looks like God gave the animal kinds a greater ability to diversify. And you can see that with their diversity. And that makes sense, because according to Genesis, God would have created not just 2, say, dogs or 2 cats, but he created them in bulk. He created population. So yeah, that's a good point. It's so funny because the evolutionists, to explain the low genetic diversity in human beings, they've resorted to that out of Africa population bottleneck 70,000 years ago to reduce genetic diversity. But what's funny is, because you were talking about inbreeding, the evolutionist explains all these DNA differences as the result of mutations. So even before the evolution of Homo sapiens, you had Homo erectus evolving independently for millions of years. Before that, you would have had your Australopithecines, for example. So there's been millions and millions of years of evolution, according to them, which means millions of years of mutation buildup. So by the time, according to their model, you get to that out of Africa population bottleneck, Homo sapiens at that time would have already accumulated so many mutations, most of them being deleterious. Now they've been decreased to, some say 2,000, others say 10,000. And then they're experiencing this bottleneck for not 1 generation, not 2 generations, but thousands of generations of inbreeding, these mutations that are recessive from the millions of years of mutation buildup. They're now coming to the forefront. And we're supposed to believe that, this highly degenerated population of 2 to 10,000 is what suddenly exploded in all parts of the world. It's nothing but fairy tale with what we know about mutations, for example. So good point.
But something else. Sorry to interrupt you, Jeremy. I should say this before I forget. I'm getting pretty tired. One of the things that has always I came across relatively recently, and hasn't really been researched heavily until the last several years, is the whole impact of short tandem repeats in the genome that they're discovering are A, not junk, and B, directly enable the flexibility of different genes to interact with different regulating factors. And some of these are only 3 or 4 base pairs, but they directly impact the physical ability for these genes to be expressed. And so to me, when you really think about the potential for genome decay, if, we've already identified, point mutations inside of a gene can be extraordinarily detrimental. Now we're discovering that point mutations in things that aren't even genes can be extremely detrimental and create massive frame shifts and all sorts of other issues to arise. And so to me, when you think about, all the arguments for in neutral theory or near neutral theory, if you actually think about it, they're mostly looking at, was there an error in, one of these high level points inside the genome, when something as minimal as something that's not even a gene or a regulatory factor can impact things from a massively negative perspective. To me, I find that very interesting. Sorry to interrupt you there, Jeremy. Go ahead, man.
It's okay. Matt, I have two questions. Just quick, since you're leaving. Does this genetic drift that you are referring to exactly identical with the genetic drift in humans?
I think Matt may have had to take off. Yeah, he just texted me. He's out of the house.
Oh, no problem. Actually, I did kind of want to comment on what you said, John, because I find that amazing too. Oh, sorry, go ahead, Jeremy. I don't know, because Matt's not here to ask those questions, but if you wanted to comment and answer them yourself, go ahead, brother.
Mr. John, so Matt is no longer here?
Yeah, he was having to leave the house.
But he's still there. He can hear us. Matt, are you there?
No, he's not on anymore. He just texted. He's not even at his house anymore. He just texted. He had to leave to go run an errand.
But John, you're very familiar. You fully understand what the raw Matt was talking about.
The majority of it, yeah.
All right. I'll ask you instead two questions. I wish Matt is still there. First question. Does the genetic drift apply that Matt is talking about exactly identical as how we explain genetic drift in humans? Because I see some differences.
Are you talking about genetic drift or genetic entropy?
Genetic drift that Matt is talking about, as it relates to these the samples he made.
SFT, you might be better at answering that one than I am.
So are you talking about the genetic drift in the species and how the reduction of variability is what is observed from the arc, changes and shifts from heterozygosity to homozygosity? Is that what you're saying?
No, not exactly. But never mind. I'll just go for the next one. I wish Matt were here. I read this from Dr. Francis Collins' book a long time ago. He said that the concept of genetic drift that I just heard from Matt, because that is what I need Matt to answer this. He talked a lot about this a while ago, and I could not ask question because according to Dr. Francis Collins, the smartest man of the world, they say, genetic drift, okay, by our standards, our model has been overshadowed the correct English, overlap whatever, by a much more familiar concept. Not much more familiar concept. It's actually natural selection, he said.
Natural selection will say it's the theory of neutral evolution, that those mutations that they assume are completely neutral. That's why they like to look at the neutral variation, because natural selection doesn't act upon the neutral mutations, and then they drift.
Yes.
they drift into new populations, and natural selection. Those mutations, they'll say, are in the junk areas, and since they're neutral, they're almost like a hidden reservoir of evolutionary advancement, that natural selection can call upon those neutral mutations, that variation down the road. So, but they're assuming that those mutations are absolutely neutral and that they can actually lead to evolutionary advancement. So that's kind of where the real question lies. Sorry, go ahead, Jeremy.
Yeah, I'm sorry. Because the design model of the evolutionary creationist and the young Earth creationists, they got their own design model too, and we got our own design model. They say to explain the design of nature. I wish really Matt were here. To explain the design we see in nature, they rely heavily on natural selection and not genetic drift. This is something I want Matt to answer. While natural selection, we know, results in adaptation. Okay, adaptation.
Right.
Genetic drift does not.
Well, natural selection has to act upon those neutral mutations that have been accumulating throughout evolutionary history and that have been drifting into other populations, for example. So ultimately, their driving forces are natural selection acting upon random variation, random mutations, but those mutations and that variation have been built up through the evolutionary process and the accumulation of neutral mutations over time. So I think they need the mutations there for the variation, then they need the natural selection to act upon that variation. But for us, we accept natural selection. Natural selection preserves species, keeps the species as strong as they can be. It's all. I think a better way to refer natural selection as differential reproduction, who's reproducing the most, but we don't assume that the vast majority of DNA differences are the result of mutation, though, as you know. So natural selection is acting upon created variation, right, according to our model. So, yeah, go ahead.
Yeah, I'm sorry. Sorry for interrupting. I'm just really I really want to ask Matt about this, but it's okay. You can.
I'm not sure. Yeah, so our model, as you know, Jeremy, we believe in, because genetic drift is just changing so if you have a gene variant, right, an allele in a population, that allele changes in frequencies due to shifts in populations and organisms, for example. But a lot of those variants, they'll say, are being built up and kept within the genomes as we said earlier, as like a hidden reservoir for evolutionary advancement in the future. But that's perfectly consistent with our model. Let's say you have a population of horses, or let's say a population of rabbits. Some have brown fur, others have white fur, with brown fur being the dominant allele, for example. Well, those shifts can change based on genetic drift. Frequencies in populations can change over generations due to chances and things like that. So I'm not sure what's your question exactly. I think we both explain genetic drift the same, but they just assume genetic drift is always about mutations, neutral mutations that have been being built up. Does that make sense, Jeremy?
More or less, but I really have to talk to Matt about this because I just see something. I just sense something inconsistent. That's all. But it's not really a contradiction. But because evolutionists relied on natural selection,
right,
but not genetic drift. Okay, and while because Matt is really emphasizing genetic drift, while natural selection results in adaptation, as we know, right. and genetic drift does not. Genetic drift does not. Okay.
Genetic drift.
So it is not responsible for those for some of those anatomy. Sorry for my English. Those not responsible for those anatomical, what else? Physiological. Sorry, English is my third
Physiological.
language. Physiological and I remember this from Dr. Francis Collins' book. Okay. Uh, and behavioral features of organisms that equip them for reproduction and survival. So this is a kind of like a contradiction to what a small contradiction to what Ramat said. So I need to.
I don't see how it's all genetic drift. It's a mechanism. It's an evolutionary mechanism where allele frequencies in different populations simply change, and they'll say, and it seems to be what we observe. These changes are due to chance, and they'll say that this is what can lead to evolutionary advancement. And
well, to be honest, to interject here, the more I am learning about some of the the newer things that they're discovering, really, in the last
Mr.
decade.
John, sorry, Mr. John, what do you think about my little objection to Roman from Dr. Francis Collins' data? You know Dr. Francis Collins?
Yeah, I've read his books. I honestly think that Francis Collins, in spite of his relevance in the Human Genome Project, I think when you look at from reading his books, I've read both of them last year, and it was interesting to me. Many of the points that he was making, I know are being directly contradicted in more recent
right. I agree with that.
But yeah, where I'm going is I'm starting to come to the conclusion that the entire premise of natural selection is actually false. When you look at the pre-programmed model, the supposed adaptations are either a modification to an existing function, or are the result of a breakage in its existing function, but the capacity for whatever the adaptation was already existed.
Sorry, may I interrupt just a sec? When you say breakage, you're talking about mutations?
I'm talking about mutations, but I'm talking about things such as the E. coli experiment and the whole glucose sucrose. Well, the bacteria had the capacity to do both. The off switch in the regulatory component of the gene that managed that was broken. So instead, it only had one option turned on. That was nothing new. It was just that the only one they needed was, I forget whether it was glucose or sucrose, whichever one it was, but they only needed that one because they were in the test tube. So the other one ended up being just completely removed. And but where I'm going with this from a much bigger perspective is, the more we're discovering about chromosomal rearrangement and epigenetics and different things that, so many of the, on the surface level that appear to be these, massive, sometimes minor, sometimes major, quote unquote adaptations, we're finding out that there was no mutation. It's literally a turn up or turn down of existing genes. And, as this is being well recognized in biology, that this, it's one of the biggest questions they're having now and SFT, that screenshot I sent you earlier about the grasshoppers and the locust.
Right.
That was a mind bender. So, Jeremy, something I came across today was that the grasshopper and the locust are actually the exact same thing. The grasshopper can turn into a locust inside of a couple hours or a day or so, based on environmental factors. There is literally no modification to the genome. The genome stays the exact same. They've mapped
So
this. And
I'm sorry.
it takes on this entirely different form through chromosomal rearrangement. So again, this takes it back to the whole point I'm making of what is natural selection?
Right. So are you saying that so I guess if you look at our epigenome, it can obviously, based on these examples, it can influence and influence the physical appearance of animals, insects, for example, in much the same way as our genes do. But as you said, John, there's no change to the underlying genetic code. It's just an epigenetic and environmental related change. And what's natural selection really? Does natural selection mutation?
Another example of this would be the caterpillar to butterfly. The genome of the caterpillar doesn't change,
right,
when it comes to butterfly, and yet it takes on an entirely different morphological form, right? I mean, there's not too many things that are more dynamic than
that's amazing.
Caterpillar to butterfly, yet the genome stays the exact same other than chromosomal modifications and epigenetic factors, right?
Yeah.
So to me, when you think about that, it's like, okay, evolutionary atheist, you're arguing that morphological changes somehow are proof beyond all doubt of evolution from your natural selection and neo-Darwinian point of view, which relies on mutation, yet we are directly observing massive modification without genome change.
Right.
How do you explain this? I mean,
how do you exactly, other than pre-programmed information, pre-programmed ability to change and adapt based on environment? Oh, sorry. Go ahead, Jeremy.
I was lost. Mr. John, does this have to do with sympatric speciation? What you're talking about?
Sorry, with what kind of speciation?
Sympatric.
No, John's just explaining how a lot of the preconceived ideas of the evolution paradigm and the evolutionists themselves about mutations, natural selection, acting upon random mutations and variation, for example, genetics and these fields of science have advanced to the point where a lot of these changes in morphology, a lot of these physiological related changes, these adaptations are simply not turning on and off of genes, the environmental epigenetic related changes. For example, like we've talked about before, a lot of those non-coding RNA genes, they're not active at certain parts in our life or certain stages of development because they need to be turned on based on an environmental trigger, for example. Is that more or less what you're saying, John?
I don't think so. Not much.
I am saying that. But I'm saying that, yes, the information exists, and yes, there are environmental and social factors that can turn them on and off. But in the context of the evolutionary worldview of natural selection, the natural selection is saying that the nature is selecting this new component that's coming into the equation. And I'm arguing that it is literally the exact opposite.
The component's already there.
The component is already there, and the variability, the capacity for the variability is already in place. Except in cases where they have relatively recently discovered some of these, portions of, supposed, duplicates and all these kind of things. They've been starting to observe in relative real time proteins going and grabbing a portion of this duplicate, rearranging it, and creating a gene on the fly. And when you think about that, one, that is a controlled function, right? Controlled process. Two, there is nothing slow about that. That's being done in real time based on an environmental pressure. This applies directly to the that's why it always cracks me up when the atheists try to use the antibiotic resistance as a supposed evidence for evolution.
Right.
It's like, do you guys actually know what is occurring in the context of how that like our immune system, for example, adapts. It takes an existing protein function or protein sequence, basically runs it through variables to find a outcome that can fight against this specific pathogen in relative real time. I mean, that is obviously controlled, not random,
Right.
not accidental. So yes, hey, cool, we've got a new ability. We got ability, to adapt to fight, XYZ virus, but that has zero relevance to undirected process. It's clearly directed. We're observing the direction occurring to result in this outcome, which is why I go off on my tangents about the artificial intelligence component of cellular biology that we're trying to copy in our AI systems. And to me, that directly flies in the face of neo-Darwinian, the neo-Darwinian model in and of itself. And then secondarily, in my opinion, it showcases the astounding level of intricacy and high level technology that enables, on the biological level, that enables us to live and survive.
And like you were saying, these types of adaptations, these types of changes, they can't actually be used as evidence for large scale bacteria to biologist evolution because that information is already present and stored in the genome. I see it as when I look at these changes, it almost screams pre-programmed or pre-existing algorithms, right? These organisms are changing and adapting based on pre-existing algorithms, as if the designer, God, of course, engineered this pre-existing information into our genomes, into the genomes of living organisms in compressed form. And now through various processes, environmental related processes, with the antibiotic resistance or Lenski's bacteria, for example, oftentimes pre-existing systems will break in order to adapt as well. But later, this information that's compressed, I would say, in our genomes, it's later decompressed due to these various processes. And what's revealed is adaptive change. But this is all just pre-programmed organismal adaptation based on the environment, for example. So none of those changes that the evolutionists look to, and like you, I laugh every time they use antibiotic resistance. Well, really any of their so-called beneficial. Sickle cell anemia, I can't believe that they use as well. I mean, that's a broken cell, broken protein, broken gene, and nobody wants sickle cell anemia. Nobody wants malaria either. But yeah, these can't honestly be used by the evolutionists to as supporting evidence for universal common descent, large scale evolution. It's just, it's mind-bogglingly complex. It really is.
Yeah, I mean, it's like saying, hey, I've got a you've got one of those multi-tool devices, right? You've got the screwdriver and the knife and the saw and all that kind of stuff built into one piece, right? And the pliers and everything all in one. It's like saying, cool, I had all these options, but I only wanted pliers because I was never using the other ones. So I broke them off. And now I'm going to argue that, oh, wow, look at those pliers. It's like, cool, I already got pliers, yo. Now I have 32 less tools available to me. And you're going to argue that this is an improvement when I had, all these different pieces that could, or a different tool set, a part of this tool set to use for, oh, cool, I need to open a letter, or I want to open the, cut the tape off this box. Well, dang it. I snapped off the knife blade because I wasn't using it. I mean, that's what's being argued by the evolutionist.
Right.
I mean, it's the not just the figurative, but the literal equivalent of what is going on inside of the degradation of the genome overall. And as Matt was pointing out on the the higher level chromosome to lesser chromosome source, it's like, cool. So now the one that's 3 layers down from the top level one, A, can never reproduce the ones that came above it, and the number that it can produce is less than the one that was above it. I mean, I'm not really following how you can come to any conclusion other than this is a obvious degradation of potential functionality.
Well, like you said, they're assuming that plier was added. It's something new. No, it was already there. You just lost 12 or 13 other options to reveal the necessity of that plier, for example. And it's like, and then I'll let you go, Jeremy, before I lose my train of thought, because this applies to John's point earlier on the Lenski experiment, because I often find, and when I debate evolutionists, for example, they define fitness in such a narrow sense. But I always point out that the best definition of fitness is total functionality. So yeah, when you look at Lenski's experiment, for example, I think he had 12 evolving strains that have been looked at, and there have been some adaptations seen because his bacteria, they're in that artificial environment, right? So they can lose genes for short-term adaptation, for example. But when you actually look at the total functionality of Lenski's 12 evolving strains, there has been a reduction in total information. Like, they are experiencing shrinking functional genome sizes. So yeah, in a very narrow sense, there's been some adaptation. But when you look to the fuller meaning of fitness, literally every single one of Lenski's 12 bacterial populations, E. coli bacterial populations, they've actually experienced what is a net loss of not only function, but a net loss of total functionality. And in the real world, you throw these bacteria into the real world, they're lazy, they're handicapped, they're functionally compromised. And Brother Sal always uses that term, and I think it's perfect, because typically that's what we see in these evolutionary type experiments. It's what we see in the wild as well. It's not just in the artificial environment. It's functionally compromised is what we see. And this is all in line with genetic degeneration and the fact that degeneration oftentimes does lead to adaptation, but that adaptation is always based on the pre-existing information. It's already there. It can't be used as evidence for fish to fisherman evolution. And it's mind-boggling how anybody could hold to that paradigm based on what we know, based on how much science has advanced in these fields.
I actually forgot what I'm gonna ask Mr. John here, but as I recall from what I read from Dr. Sanford's book a long time ago, I didn't fully read his book, but I read most of it, and the evolutionary data as well. As we know, genetic entropy usually from heritable disease, correct? Right?
Very often, yeah.
Yeah, very often. That's because protein coding. Sorry for my English. That's because protein. Sorry, that's because protein coding are less tolerant. Okay, they are less tolerant. Is it tolerant? Yeah, they are less tolerant of the mutations from other parts of the DNA.
Absolutely. I mean, no, the interesting thing though, what they're discovering now in research is, and yes, you're 100% correct in regards to, very minor mutations, and that protein doesn't work anymore. It's done. Yes. What they're also finding out, especially in cancer research, is that a significant amount of cancers are directly caused by some very often extremely minor mutations in non-coding regions of the genome as well. So it's that error in the regulatory function causes a cascade error to occur further down the line and mess up a bunch of other factors, one of which may be a gene that now goes haywire because of a very minor um, mutation in a non-coding regulatory region, and which is why the entire premise of so much of evolution, the evolutionary arguments are dependent on the majority of the genome being non-functional and being evidence for evolution. And obviously that entire premise is being destroyed by more recent discoveries. But as they find out more and more components of the genome that they thought were completely worthless are directly in control of whether or not we have a disease. I mean, that entire position that they're taking is collapsing around them. And yet so many of the most staunch advocates for evolution are doubling down on the supposed junk DNA and the their standard natural selection and adaptation through slow mutation, which is just, I mean, there's a rapidly growing chunk of researchers who are even on the evolutionary biology side that are starting to raise their voices about how this is stupid and this is like a pointless, like, why are we following this model? And then on the medical research side, there is a very large percentage of people doing the medical research who are basically telling the evolutionary biologists to go away because what we're discovering flies in the face and is directly contradictory to the positions that you guys are taking. We're discovering the exact opposite of your argument about evolution. So just go away and leave us alone. Stop getting in our way. We're actually behind the times on what could have been discovered and different diseases we could have just figured out how to treat if we hadn't been listening to you idiots for the last 40 years on what we should be looking for in terms of disease and just healthcare in general. And to me, when you really think about that in the context of what have we been focused on? We've been focused on no more than 2% of the genome for at no less than 50 years. Focused on 2%. 2%. And yet we have these people arguing that we just know so much. I'm like, really? We know a whole bunch about 2%. That is not there's not too many other contexts where you could consider yourself an expert if you only knew a portion of how 2% of something worked. You would never be considered an expert at that point. And yet anybody who presents counterpoints and counter arguments to their models is somehow considered out to lunch. And all of us talking tonight, we're just a bunch of crazy young Earth creationist religious types who are Bible thumpers and aren't using our brains. And it's like, when you actually look at what's being discovered, it seems that you guys are the ones that have been extraordinarily closed-minded, not people who have been in disagreement with your so adamant and dogmatic positions.
Mr. John, going back to thank you for explaining. Going back to what I said a while ago, as we agreed, of course, most of the genetic entropy are usually from a heritable disease, right? That's because, as we know, protein coding are less tolerant of those mutations than other areas or parts of DNA. Now, as the genetic entropy decreases, these tolerance levels would also decrease.
Right.
Correct? Right?
Yep.
Okay, so thank you.
You back standing?
Oh, yeah. Well, and the thing is, they're finding so much with cancer research and the things you were mentioning, John, that so many of these diseases are now attributed to the non-coding regions. And typically, you don't even need to know exactly what the function is. if there's a disease associated with it, for example, there's diseases like RJ Downer always says, well, these ALUs, sure, some of them are functional, but some of them are disease causing. But it's like, yeah, of course, if you have a functional DNA element that experiences a mutation that's detrimental or just deleterious to some extent, you would expect the result to be disease, to be genetic damage. But yeah, if you're familiar with Grar, he's got that paper recently, and Brother Sal talks about it. And Erica, in my second debate, thought that it was like the fatal blow, right? Because Grar tries to put a cap on the amount of function in the genome. And it's because evolutionists have been so triggered by ENCODE. Once again, as Salvador always points out, Grar is the one who said if ENCODE is right, then evolution is wrong. And we know that ENCODE has revealed at least 80% of our DNA is saying functional itself may be premature, but it's at least active. It's doing something. What is every single sequence doing? Well, we understand so little of the DNA language that it's going to take years and years and years of experiments and knockout tests and stuff to know exactly what every single sequence is doing. But it's there for a reason. It's being transcribed. The cell's not going to transcribe junk for millions and millions of years. That would be just wasteful of energies, for example. So the more functional our genome is, the more the genetic degeneration issue, it's more of a problem for the evolutionists now, because they can't explain genetic entropy away with junk DNA or those non-coding DNAs that absorb the mutations, and they're held for evolutionary advancement in the future based on genetic drift and things like this. So that Grar paper, it's funny because he says that for human population levels to be sustainable, as in to counterbalance the effects of harmful mutation, and I've read over the paper since these evolutionists are all using it now, and he concludes that no more than 25% of the human genome can be functional, even though ENCODE, as we said, has revealed that at over 80% is at least active. So he has to conclude that ENCODE is completely wrong, or else evolution can't be true. But the funny thing is, when you read this paper, Grar is trying to get around these issues by, and here's the ridiculous point, he's assuming deep time evolution to prove that the vast majority of the genome is junk, because he's saying that since we as a human species has been around for a very long time, another assumption, and since we are not extinct, then it has to be true, because evolution has to be true. They never question the basic assumption of universal common descent. So he concludes it has to be true that these mutations are striking in the junk portion of the genome. And this is obviously circular reasoning, which is hilarious. And I love when the evolutionists use that paper. So he's saying, and I think Salvador explained this a couple lectures ago, but he's saying human females cannot have enough babies to provide enough fodder for natural selection to remove all those harmful mutations from our population. So the only solution, in his mind at least, is that the majority of our genome is junk. But what's funny is he just won't question the fact that maybe humans are not as old as evolutionists say we are, based on the function and the deleterious mutation accumulation. I know I went on for a while there, but I was
going to make a quick interjection on your point there, man. And I'm going to step away for a second after this. Something that this admission that I'm about to reference, I get dramatic amount of entertainment out of it, but more importantly, in regards to what you're talking about, SFT, what is functional, right? Well, they point blank admit that there is dramatic difference in vivo versus in vitro research they're doing, right? They're starting to recognize beyond doubt that there's all sorts of stuff that happens differently between the two, right? So if we're not even doing research on very heavily minimized research, right, in terms of in vivo versus in vitro, right, we actually don't have a clue of how much of the genome is actually active, since so much of it is, well, I mean, all of it is dynamic, right? It operates on the fly based on a specific scenario. So there's all sorts of portions of the genome that are there more than likely, and they're discovering this in other cases. Oh wait, oh wow, this one only operates in XYZ scenario. Well, if we're we're never going to be able to study every possible scenario in which a human could possibly be in regards to how a portion of the genome might operate in that specific context, because we're not in the lab. And to me, when you think about that, it's like you guys are operating in a serious state of denial of, I mean, that's everything I just said there is not that complicated. Like that's a pretty basic observation, right? That you guys admit yourselves to be true. Yet the staunch, advocates for evolution, like they dismiss that premise in terms of whether or not portions of the genome are functional. I'll be back in just a minute, guys.
Well, I guess John's probably not going to hear what I'm saying, but yeah, it's funny. It's flabbergasting to think about how many experiments, how many thousands and thousands of experiments would actually be required for any scientist to conclude with any level of confidence that a certain set of DNA sequences in our genome has absolutely no function at all, for the main fact, as John was explaining, certain genes function in different ways, in different stages in our life, different stages of the developmental process. Think about the incredibly complex sequence of events, okay, that changes a single-celled egg during development into a fully formed organism with hundreds of different cell types in different organs. They have to all be in the right places. And guess what? This programming to take that single-celled egg into a highly complex organism has to be somewhere. And there's good evidence that suggests that it's within the areas that the evolutionists say are junk. But to test for every single non-coding RNA gene, for example, or pseudogene that's involved in these various stages of the developmental process, the number of experiments that would have to be done are just unimaginable. Jeremy, what do you think, brother?
Yeah, I agree. Evolutionists would say that we generally. Which part do you want me to address again? Standing for truth.
Oh,
I agree with what you said, but I would. Are we gonna make this comprehensive? Are we gonna use the evolutionary argument also?
yeah. Why don't we go into some of the evolutionary arguments against genetic entropy? Well, I think we just thoroughly refuted their argument that the majority of our genome is junk, because they're going to assume it's junk, just like that Grar paper, in order to make those deleterious mutations absolutely neutral. But because of the function of the genome, we know that those neutral mutations are just, a lot of times they're slightly deleterious. Therefore, but they're still neutral, nearly neutral. So natural selection, essentially they're invisible to natural selection. They're going to build up. They're slowly going to harm and degenerate our information system. So I think we thoroughly refuted that one. What other arguments are you thinking, Brother Jeremy, that the evolutionist uses?
As I recall, because I am not really updated with this, as you know, genetic entropy is yours, chromosomal 2 fusion is yours, and the rest are ours, and some are mine. Okay, so we have our own fields in genetics alone. Now, sorry for prolonging it. As I recall, some of the evolutionary arguments, like synergetic epistasis, something like that. But I'll go to that later. Evolutionists say that we cannot, as I recall from the peer-reviewed argument against genetic entropy, evolutionists say that we cannot generally see uh, measurable, that we can something that we can measure, or measurable degeneration in extent when we experiment it in a lab, in an extended lab experiments. Okay, then they would also, let's say, that in the last few thousand years, uh, uh, all forms of life having short life cycles, like bacteria, okay, and even other examples like mouse or something else, they should have gone extinct. Since we do not see obviously degenerating happening, the what you call it in English? This is a deep English for me. The primary axiom. The primary axiom must be true. So that is one of their counter arguments. You want more?
No, yeah. Did you want me to address that, or did you want to address it?
You could address that. I know how to address that, but you could do it because you are the expert on this subject.
Sure. Yeah, no, that is a common objection. That's why I don't want to rehash Lenski's experiment, but he's shown clear evidence of genetic degeneration. Michael Behe goes into that in great detail in his new book, Darwin Devolves. But the fact is, and mice, for example, like Matt and I showed that paper earlier, that mutation rates. Now, generation times change. Don't get me wrong. Mutation rates in mammals, for example, are generally consistent. Now, evolutionists always harp on us, that we assume constant mutation rates. Yeah, we do. I prefer to say consistent, because there may be minor fluctuations here and there, but generally they're consistent. They're not 10 to 20 to 30 fold different in the past, like the evolutionists say. Now, that's not consistent. But we generally assume consistency, which is why we can make testable predictions. We're doing the science, for example. They have nothing more than post hoc ad hoc rationalization. But the reason why mice still exist would just be that there hasn't been enough time, just like with humans, to for them to go extinct. But with bacteria, and mice, of course, have higher generation times and larger population sizes, but genetic entropy has never predicted that bacteria should go extinct before humans and higher mammals, for one. So when they use that argument, it's kind of like a straw man. But what's fascinating is, as I said earlier, Lenski himself, he's not even doing it on purpose. Those bacteria are degenerating. They're losing genes. You can see shrinking functional genome sizes. So it's genetic entropy at work. But for the most part, yeah, bacteria, they're simple. They have simpler genomes. Population size is a major consideration. You have thousands and thousands of bacteria just in your body alone. And bacterial species, just like viruses, they're also known to undergo major periods of dormancy. Even if some bacterial populations went extinct here in North America, I mean, there's just so many trillions and trillions and trillions of bacteria on the face of this earth. They would never go extinct, but we can look in the lab and we can study it like Lenski is, and we can still see that they are prone to reductive evolution. But yeah, bacteria, for example, they have short generation times and lower overall mutation rates. What this means is bacteria will have more efficient and more rounds of natural selection. Because of how simple they are, if there's a mistake, natural selection is going to see it. It's kind of like if I have a tricycle. There's really only a few parts. So if something breaks down, it ain't going to work. I'm going to notice it. I'm going to fix it. I'm going to have to act quick. Let's look at a higher mammal like a human being. Picture a human being like a Chevy Camaro, or like, what's your favorite car, Jeremy?
My favorite car, Vios.
Beetles?
VIOS.
Okay, so a Vio. So picture how incredibly designed and complex that car is. Little things can go wrong in that car, and the car can still work properly. There's a lot of times where people go, unfortunately, they go a long time without bringing their car for a tune-up, and a lot of times problems will build up before something actually breaks down, where they're like, oh no, I got to take this in to get fixed, because there's more things to go wrong before we may notice, versus the tricycle, which I'm comparing to a bacteria, where one problem, boom, the tricycle, the bicycle, whatever it is, it ain't going to work. Therefore, it's easier to see. So natural selection works better on bacteria. So yeah, overall, bacteria would be predicted based on the model of genetic entropy to be the most resistant to genetic entropy. But that being said, what I find amazing is we still see paper after paper that goes into great detail of bacteria that are degenerating, losing information for short-term gain, for example. Yeah, so I think that because typically they'll say, yeah, if genetic entropy is true, why do bacteria still exist? So I gave a number of reasons, a number of ways how you can address that, but I always emphasize looking to their number one experiment. Richard Dawkins himself, in his book, The Greatest Show on Earth, goes into he goes into great depth in that experiment. But yet that experiment proves devolution and not evolution. So yeah, generation times play a huge part, and population sizes play a huge part as well. So good objection, Jeremy. Hey, what are your thoughts on that, brother?
That is why mutation rates are also used against the genetic entropy objections, right?
Right.
Okay. Another rescuing device. You know this too, as I recall. Synergistic epistasis. Okay. Based on what I read from Dr. Venom's book, it really happens. And based on other peer-reviewed papers that I read, which even our neutral evolutionist who doesn't even care about the creation evolution debate. Yes, I would admit that some forms of synergetic epistasis really happens, at least to some small insignificant level. You agree with this standing for truth? Before I continue,
I do agree with that.
At least.
Jeremy, real quick, hey, did you want to explain to the audience what synergistic epistasis is, just so they know what we're talking about?
It is the rescuing device for genetic entropy, and
yeah. Did you want me to explain
least,
like what it? Do you want me to explain what it is?
Yeah,
like,
you could explain it. You can explain it.
Okay, so they have two main artificially contrived mechanisms. One is synergistic epistasis, like you're saying. Another one is mutation count mechanism. Okay, synergistic epistasis is okay. The best way I could explain it then, if we have lots of mutations, okay, that are deleterious. So these nearly neutral mutations that accumulate that we're saying are invisible to selection, and on their own, individually, they are inconsequential. Okay, but it's the buildup of them over time that degenerates our information system. So if we have lots of these mutations, they're deleterious. As they accumulate, this is what synergistic epistasis suggests. Each mutation amplifies each other's negative effects. Okay, so now the intensity of degeneration based on this amplification is so powerful that it is at this point where natural selection can see the damage and act upon it. Now, yes, it as Jeremy said, it does work to some extent, but the problem is, and this is what logic would dictate as well, using and invoking synergistic epistasis to solve genetic entropy actually makes the problem worse, because it just leads to faster extinction. okay, this population has so many deleterious mutations accumulating that it reached a point where they've all amplified each other's effect, where now natural selection can work. Boom, gets rid of those highly degenerated organisms, but it doesn't do anything to fix the problem overall. And it only makes the problem worse. Like I said, faster extinction, faster genetic entropy. But most importantly, highly extensive numerical simulation experiments. And there's technical papers published on this that I could never go over sufficiently in the span of time that we have here. But I would invite the listener to read them. I've read them. They're great reads. But these numerical simulation experiments, they've actually falsified that mechanism of synergistic epistasis. So you can actually see this in print. And oftentimes in these settings, they will invoke the most generous of settings. And still, what they find is synergistic epistasis, although it does happen once in a while, as Jeremy said, I agree with that. It always fails to stop or prevent deleterious mutation accumulation. So another thing, and I learned this from Salvador the other day in his debate with Ron, even Grar, the one who's so opposed to ENCODE and genome functionality, he doesn't even invoke synergistic epistasis, and he's fighting it so hard. He's looking to genome functionality and trying to say that most of it is junk. So yeah, these mutations accumulate, but they accumulate in the junk areas. That's what he's looking to. He doesn't even look to synergistic epistasis. So, you know, the evolutionists that do, they're just behind in the times, and it's already been falsified. So, but that for a time being was probably their best rescue device. But what are your thoughts, Jeremy?
Actually, as I recall from one peer-reviewed paper on the sorry for cursing again. The synergetic epistasis.
No problem,
okay. Most of their peer-reviewed papers do use magic words regarding the synergetic. Not really a magic word, but most of them, those peer-reviewed papers, just say that this might,
right?
This probably, or this could. Okay,
right.
not sure. Aid in mutation, and aid in mutation elimination. Right.
Right. Good point.
Yes, and
okay. So you're saying that even they're admitting in their own papers that it may or may not. They're not even sure.
Yeah, if you read their papers, they are not sure.
Right.
Okay. Yeah, they are not sure. So there's another explanation for synergetic in their synergistic.
No problem, brother. It's not an easy one to say, anyways.
My English is third language. Let's say when the effect on fitness of two mutations, let's say, is more what's the English again? I know the Chinese of this, but English. Okay, when the effect on fitness.
Oh, actually, real quick, Jeremy. I'm so sorry. I'm gonna let you continue. Brother Nephilim Free, awesome, because he saved me a trip, saved me some work. I was gonna go post the paper, that one of the papers I'm talking about. So he posted it in the chat. So I recommend anybody look into it. 26 pages. It's titled "Can Synergistic Epistasis Halt Mutation Accumulation? Results from Numerical Simulation." So thanks for that, brother. Matt.
You wrote it? You wrote the paper?
No, he didn't write it. He although he is smart enough where he probably could, but no, it's John Baumgartner, Dr. Sanford, Dr. Brewer, and it's www.worldscientific.com. So thanks, brother Neff. He posted it in the live chat for people to see and save and read. So
because thank you.
Go ahead, Jeremy. I'm sorry to interrupt, brother.
Yeah, it's okay. Okay, so I don't know if this is a easier way to explain it for the audience. When the effect on fitness of two mutations, okay, for genetic students out there especially, is more radical than finally I know the English, radical than their effects when alone. it is referred to synergetic epistasis. And there's another epistasis that I remember. It is called anagonistic epistasis. It is the opposite situation. When the fitness, okay, of the double mutated, gene from a wild type, from smaller effects from the effect of two single mutations. Yes, that's called an agnostic epistasis. Okay, so mutations that are being deleted, deleterious mutations, negative epistasis is also uh, uh, synergistic. Oh, synergistic. Okay. My goodness, I don't know how to explain this in an easier way. Uh, yeah, synergistic. Okay. Okay. While these positive um, epistasis is antagonistic, like I said. Okay. Okay. For beneficial or advantageous mutations, positive epistasis is synergistic, when negative epistasis is antagonistic. That is from a peer-reviewed paper that I've read a long time ago. What do you think, my friend?
Sorry, brother, I was on mute. No, I think that's awesome. I think I'm glad you're bringing up these tough objections, because it shows it helps us to be as comprehensive as possible, even on one topic of genetic entropy. This is how detailed we can be. So you're right. There's those two mechanisms. There's one other one called mutation count mechanism, which says
anagonistic epistasis.
Which one?
Anagonistic epistasis. This is the other one.
Okay. So there's those two types of epistasis. There's also another
sign epistasis, as I reckon.
Okay, perfect. And there's another mechanism they look to as well, which we can call mutation count mechanisms. But this is another one that simulations have disproven. This one's not even biological.
Sorry, standing for truth. Who was disproven?
It's called mutation count mechanism. So you know what? You're right. I should explain it. So what this mechanism says, okay, as mutations accumulate, Mother Nature is then able to count the number of mutations per person, and then Mother Nature, natural selection, can select the higher number of individuals. But that one's even way worse than synergistic epistasis, because it's not even biologically real. The model itself doesn't even resemble biological reality. But most importantly, numerical simulations, just like synergistic epistasis, they have shown that even this mechanism fails to halt deleterious mutation accumulation. So, and it's just like the synergistic epistasis, like you said, Brother Jeremy, when they talk about it, it's more so, here's the thing. Evolution is true to them. They don't question the theory itself. Little details within it they'll question, and they'll write papers and fight over, but they don't question the actual theory itself. Therefore, to them, evolution happened. So, what mechanisms can we invoke? What stories can we tell? Pull out the storyboard. Let's figure out how we can halt mutation accumulation. And that's why you'll often hear the stories. You'll see it right in their papers. You'll see the maybe this, maybe that. But the observable facts are genetic degeneration exists, it occurs, and there's no way natural selection. Oh, here's Brother Neff. I mean, Brother Neff was debating R.J. Downard and Jackson Wheat, and Neff pointed out that natural selection can't see the majority of mutations. Unless they're highly, highly detrimental to the organism, natural selection will see it. Or it may amplify one of those few rare beneficials, even though they're still reductive in nature, sickle cell anemia. Sure, maybe it'll amplify one or two here and there. But the fact is, those nearly neutral ones, they're invisible, and that's the majority of mutations. So they just build up, and that's why evolutionists have to say, "Oh, these are neutral," because the vast majority of our genome is junk. Remember when you debated Tony Reed? You know, he fought for that one, brother Neff. So good to have you, Neff. What are your thoughts?
Well, something that I think a lot of people don't realize is that evolutionists have to deny that the genome is crumbling because of the effects of mutation. Because if they acknowledge genetic entropy is true, then the game is over for evolution. It's completely over, and not just kind of over. It's dead. They have to give it up. Every scientist in the world has to look the other in the eye and admit evolution is not true, because evolution is the base mechanism for evolution, allegedly, is random genetic mutations acted upon by natural selection. So if random genetic mutations are not, in fact, the designer of all the fantastic things about organisms, but instead causing organisms to decay, then there is no mechanism for evolution. Evolution can't possibly be true. So the evolutionist is stuck with having to deny the mountain of scientific evidence that shows that random genetic mutations, the accruing effect of a slightly deleterious random genetic mutation, is decaying the genome and causing organisms to move towards extinction. They have to deny it. Absolutely must.
And quick interjection, guys. I'm gonna have to call it. I've been up since 5:30. I'm about to crash. The something else that I've been finding very interesting, and I think has significant bearing on what we're talking about now, is, you know, for years, in spite of the, you know, three-dimensional structure of DNA, we really approached it from a very linear perspective. And now with the different things they're discovering in terms of the very significant aspects, you know, from a 3D and a 4D perspective, you know, they're recognizing in structural biology, we actually don't know how much the supposedly neutral mutations are actually affecting the genome overall. In, you know, we've been looking at it from much more of the linear perspective, but those supposedly neutral mutations could actually be having a negative effect in it all the way up to an entirely different chromosome. To me, when you really contemplate that, of mutation A in chromosome 1 could actually be having a negative effect in chromosome number 2. Like, how do you even remotely, like, how do you identify what that is? How would you even know? Which, I mean, I'm sure we could probably figure out a way to research this, but I mean, it's been relatively recently, I mean, within that last decade, we've even started to truly recognize the significance of that. Like, correct me if you guys think I'm wrong, but we've been hyper-focused on, oh man, you know, what's going on in this genome, you know, this specific sequence, and it, you know, what's in the relative proximity to that sequence. And to now be faced with the reality that that modification could have a deleterious effect in another chromosome, I mean, to me, that's a mind bender, number one. Just from an engineering perspective, when you think about the foreknowledge requirement that was required to create a functional component in the first place, right? But then you take it to the next level, and from our perspective of, to me, it just puts a whole new layer of how much we actually don't know about genetics, in spite of the, you know, so we hear so much about how we know all these things, and any counterpoint to the evolutionary model is false by default. And it's like, you do realize we just discovered we know even less than we thought we did before, right?
Mm-hmm. Good point.
Yes.
John, I liked your video, 4 Ways to Trigger a New Atheist. That was funny. I like that, man.
Loved it. Anybody in the audience who hasn't watched that video, check and subscribe to John's channel as well. He's putting out some great material, and brother Neff too. But Neff, you're famous. Everybody's subscribed to you.
Yeah,
Actually, Jeremy,
8,000 subscribers, man. It's share the love, Neff. Share the love.
share the love, man. John's a warrior.
I appreciate you like that video, man. I was cracking up when I was writing writing down what I wanted to say in that one. I was on the back porch just cracking up as I was coming up with what I wanted to say. I'm like, man, this is this is so true. And the ultimate irony is, while I was writing what I wanted to say, one of my trigger points was I got a notification of a comment, and I was reading through it. I'm like, wow, I was literally writing about this exact trigger, and it's happening in real time in the comment section of a different video. This is awesome. Anyway, guys, I gotta pass out. Y'all have a great night. Appreciate coming together. And
take care, John.
Have a good one,
You're
folks.
always welcome.
To meet you, Mr. John.
Great to meet you, Jeremy. Enjoy it.
Since we're gonna make this video complete, more comprehensive, standing for.
Well, I just wanted to make one point off of what Brother John was saying there, because I think that is really important what he's saying, because the evolutionists assume that the set of instructions in our genetics, they assume that it's just it's a simple linear array of letters. This isn't true. It's as Neff always says, it's nested, it's multidimensional, it's dynamic, and it's self-regulating. That means more things can go wrong than can go that could get better. I mean, one single change can break an entire system. Go ahead. Just wanted to point that out.
Who, me?
No, yeah, whoever.
Yeah. Since we're gonna make this comprehensive, before we go to the mutation rates, which is very critical, there's also an evolutionary argument from a peer-reviewed paper I read a long time ago. I seldom hear this in SFT debates being opened up. But this is an argument a long time ago, and evolutionists have forgotten this argument also. But some still use it. Okay, some. Evolutionists also mislead people by telling them that some specific genetic entropy markers are just a few in the genome. They are not as big, or as what's the correct English? Not as extensive as what we are saying. Okay. Some even said that these genetic these what you call that again in English? These genetic entropy genes are just not so many, something like that, more or less like that. Okay, they say it's just a small part of the genome. Anybody want to answer that, or I'll just answer that?
Go ahead, brother. Tell us.
I'll just answer that myself. Now, which book? Yeah, when I look at the genome, yeah, it's true. In some cases, okay, it's true that these genetic entropy samples are less than they are not as many. Okay, sorry for my English. Okay, the answer will be that the small specific genetic samples are not really literally they are literally small. Okay, but even if they are small, they are critical. That is what evolutionists are not telling you. Okay, even though it's true that some of them are small or isolated, these genetic entropy samples are critical. Okay, and if these really go further down the road, if the English is correct, then it would really compromise the species. Okay, so that's their point. The genetic entropy samples that we are giving are just a small part of the genome. But what they are not saying is these small samples are very critical. That's all. We could go to mutation rates, standing for truth, or whatever.
Well, I was just going to say, even in the largest part of our genome, the non-coding areas of our genome, non-protein coding RNAs, they literally regulate all aspects of the gene expression pathway. So even if you get the vast majority of mutations, even if they're being, if they're in that part of the genome, only 2% of the genome actually codes for protein. So if almost all of the mutations are in that area, I mean, that's still going to be slightly deleterious. So yeah, I believe that genetic entropy certainly does affect. If there's a point mutation or a mutation in something that's critical to our health, well, that's going to be damaging that first generation. Heck, that person will probably die, especially in the mitochondrial DNA. I mean, detrimental mutations in there that have to do with energy production and ultimately the heart. Mutations are not generally sustainable there. Large mutations, that is, but the non-protein coding areas, I mean, the amount of function in those areas of the genome, boom, you get 60 mutations in there in a generation. I mean, those are going to be deleterious enough where they affect genotype a little bit, phenotype a little bit. And Neff, what are your thoughts, brother?
Well, the complexity of the genome is amazing. As John was talking about, gene that affects one mutation that affects one gene can affect others, and partly because of the overlapping and nested nature of genetic information, which evolutionists disliked when it was discovered. They actually didn't like that, but also because genes in one location of the genome regulate information that's expressed in another part of the genome. And so if you mess with a regulatory gene, you can screw up the cell's ability to properly use, the whole organism's ability to properly use information that's found in another part of the genome. So, you could have a regulatory RNA transcript in one chromosome affect the expression of genes in another chromosome. And so those genes don't get expressed properly because a mutation has damaged the regulatory RNA network. And so the complexity of DNA is really just so mind-boggling. It's like the idea of shooting 22 caliber bullets into a stack of books. How is that going to make the books better?
Right.
The evolutionists can't stand it. That they actually dislike it. That DNA is as complex as it is. They would really prefer that it was really very, very simple, because then it makes their evolution idea more plausible. The more complex we discover DNA to be, the more problem it is for evolution to be believable.
May I, since standing for truth, for the past 4 videos that we made great videos, right? We represent the other side very well. Shall I use a little discouraging data from evolutionists? I mean, discouraging for young Earth creationists.
Before you do that, Jeremy, let me say this. I've noticed how ardent you've been about making videos about.
Sorry, can you repeat?
I wanted to point out. I've noticed how hard you've been working, making videos, and they've been about the universe and about galaxies and about the Big Bang, and everything that's being published on Standing for Truth channel. And I just think that's awesome, man. You're a hard-working dude. I really admire the effort you're putting into making a stand for the truth, Jeremy. It's amazing. It's really awesome.
Neff, I hope you could, because Ramat and I during his broadcast a while ago, in his channel a while ago, we were talking about hopefully we could all integrate our efforts. So why don't we all unite our channels together?
Well, there's one thing that we could do. I created a channel called Evolution Review Group, and I created that site for a purpose, and I haven't tried to get around to doing anything with it. But the reason I made it was this: I thought it would be great if some of us would get together, like a peer review group, and we would have live streams from that channel. And what we could do is review a video made by an evolutionist, pick it as though it were a science paper, and we are a journal review group, and we
Yes.
could watch the video, and then we could go around the table several times, giving our opinions about the claims made in that video, and show what the evolutionist has done that's wrong, show the scientific evidence that is wrong. Kind of like a review group. I think it'd be a great way to intimidate and shame some of these loudmouth evolutionists. If you had a group of creationists, 3, 4, 5, 6 of us, going around the table, all giving evidence that refutes the claims in that video, I think that would be awesome, and we ought to get together and do that sometime. The channel, really, I created it, but it doesn't really belong to me per se. I mean, I had the login for it, but it's not for me. It's not my channel. It's a channel for us, really, to use, and I think we ought to take advantage of that sometime. That's one thing we could do.
I think that's a great idea. We do need, and we've discussed that 2 or 3 times. We do have to get on that, because this is such an important thing for us to do right now in these times. So
I'll give you the link to it, Jeremy, so you can at least see the channel. There's no videos there, just an intro video, and I'll paste the link in the side chat.
In the private chat? Okay.
No, the side chat, where the main chat. From Yeah, there it is. There's the link for it, and it's just a site for all of us to meet on once in a blue moon, have a hangout and review an evolutionist's video and show the scientific evidence that the claims they make in it are ridiculous.
Okay, thank you. Like I was saying, standing for truth, to make this video comprehensive, shall we go for the evolutionary evidence that is very discouraging, or not? It's okay if it's not. Standing for truth? Where is he?
He probably stepped away from his computer for just a minute.
Yeah.
He'll probably be right back.
Okay. So you up. Okay. So what else do you want to say, Anya?
Well, I would just say that it's telling that evolutionists fight so diligently against discoveries in science. Every discovery that adds a layer of complexity to what we see in biology, to biological systems, they actually don't like it. They despise the complexity of organic systems, because it shows the implausibility of evolution having designed it, and then when mutations, the implausibility of mutations being a designer of it, and in fact instead a destroyer. So the more complex it is, the harder it is to believe evolution. Evolutionists actually, they not only hate real science, but they hate the complexity of living systems. They despise it because it makes evolution unbelievable. And I just find that remarkable. They're actually anti-scientific. The irony is that evolutionists claim their ideas are scientific, and the creationist is anti-scientific. The opposite of that is actually the truth. It's them that hate real science.
Yes, I agree with that. Shall we go to the mutation rates?
Yeah, sure. Tell us something about mutation rates, Jeremy.
Yeah, is standing for truth there? Still not there?
He probably had to step away a minute. I think we'd know it if he was back. He'd be talking, probably.
Yeah, well, mutation rates can be used. Sorry. Yeah. Okay. Do you have something to first say on mutation rates before I do? Yeah.
Well, I mean, I could say a lot. Why don't you go ahead and take us into it and share your thoughts about it?
Yeah, I'll go for the basics. The mutation rates of DNA usually is applied in the mitochondria or the mtDNA. These mtDNA are actually small organelles that are outside the cell's nucleus. Okay. There's a design feature here. Okay. There's a design feature here in which why the mitochondria is outside the nuclear DNA, because mitochondria produces free radicals. Okay. And we know what free radicals are. So it is not contained in the primary nuclear DNA. So this is a design feature. If it is contained within the primary nuclear DNA, then well, we're dead. Okay, so this is the design feature. So again, to repeat, is standing for truth back? Are you back, standing for truth? Still not there. Okay. Um, where was I? Okay, like I said, they are small organelles that are outside the cell's nucleus. Okay, and then I explained the design feature of it. Mitochondria contain their own DNA separate from a creature's main genome. Mitochondrial DNA is actually inherited from the creature's mother, as we know, and its mutation rate can actually, accurately be measured to produce a molecular genetic clock. Okay, mutation rates, like I was talking about, by comparing rates in a few different animals or small species. Okay, and we have demonstrated that a creation of not more than let's say 10,000 years. So the max is 10,000 years ago is confirmed by these genetic clocks. So that's the basic explanation. Okay, standing for truth. Are you back?
He's still going, I guess.
Still gone.
So
Anything
yeah,
else?
the mutation rate is approximately 100 per person per generation. Every human being has 100 mutations more than their parents, and that's alarming. And evolutionists have tried to claim that epistasis is a solver of this, but of course, it's not, and it's been demonstrated by Kondrashov and Lynch and Connery and Berger and Higgins and Lowe have all published papers in the mainstream secular journals like the National Academy, the proceedings of the National Academy of Sciences, that show that it's a real problem, and it's only getting worse, and there doesn't seem to be anything that's fixing it. And so Jonathan Sanford, Baumgartner, Gibson, Brewer, Remine, and others have demonstrated with their own papers that that this is a killer of evolution theory, and evolution can't be true because of it. So, but it's an old idea that it's been demonstrated for a very long time, even by secular geneticists, that genetic entropy is true. For example, Ota in 1973, 74, and 92 and 2002, and Kamura, both are Japanese geneticists, in 79 and 83 published papers explaining that random mutations are slowly degenerating the genome of living things, and natural selection can't stop it. And of course, for that reason, they were, they became disliked by the evolutionist community, even though they were, before they published those papers, they were, the poster boys of genetics in the world. And then after they publish these papers, suddenly the whole scientific community, secular camp, dislikes them because they're just telling the truth about what they observe. And so it's remarkable that the evolutionist has to claim that natural selection is able to remove them when it's been demonstrated that natural selection can't see a mutation unless it causes a profoundly negative effect, and then natural selection could act to remove that individual and its mutation from the population. But natural selection can't see the average mutation. And that's why Condershaw published his papers asking, why are we not, what was the name of it? The degeneration of the human genome by slightly deleterious mutations. Why are we not dead 100 times over? Because he's just being honest about the information, but the evolutionist has to be in denial of the fact that mutations, the majority of mutations cause a slightly deleterious and degenerative effect, a very slight one, but nonetheless slight. And over time, this accrues, and it just causes continual damage to the genome and makes the organism increasingly less fit to survive. Evolutionists have to be in denial of this today, because as I pointed out, if they're not in denial of it, then evolution theory is dead. It's absolutely so dead, in fact, and it is dead because of this, that evolutionists would have to actually admit to each other that evolution theory is dead. And they just can't fathom that. They're not going to go there, so they have to deny science.
Well, thank you for saying that. And finally, standing for truth is back. I just like to. This will be short, and then your turn, standing for truth, and we will give specific samples because we are now at the mutation rates of DNA, which is critical, or one of the most critical. Okay, now for the audience.
Real quick, Jeremy, I just want to point out, because one thing there stuck out to me that Neff said that I think is really important. I want the audience to pay special attention to the fact that natural selection, just like Neff said, is only going to remove those mutations that have a profound, that was the word you used, Neff, and you're absolutely right. Only those mutations that have a profound effect on the organism that's detrimental enough. Yeah, those are the ones that natural selection is going to remove. But the vast majority, and you pointed out that we accumulate roughly 60 to 100 new mutations per person per individual, most of those being nearly neutral, and only having a very slight effect on fitness. So if you think about it, if we're transmitting a small amount of mutations to our children, so you got the first generation, 60 to 100 additional mutations. Your second generation, you got about 200. Your third generation, you got 300. Think about this. If every single person on this planet, okay, is multiply mutant, and if every generation then, if you're accumulating more and more and more every single generation, that means every generation is more mutant than the one before it. So all this selection can do is slow down the degeneration by getting rid of the absolute worst of the lot, those ones that have had a profound enough mutation, but it does absolutely nothing to stop the mutations from accumulating in the population over time. There's nothing, it can only slow it down. It can only help us to an extent. It's limited is the point.
Right.
Okay. Finally, we are in the mutation for rate, standing for truth.
Oh, sorry, Jeremy, we got to shut it down. Anyways, guys, have a good night. No, I'm just kidding. Go ahead, brother. Sorry, I had to step away for a bit. I was getting some food. I had to take a little break. So go ahead. So mutation rates?
You're going away again?
No, I was just kidding. So Jeremy, go ahead, brother.
Well, do not take advantage of my weak English, my friend. All right. Because I sometimes do not understand the grammars, and if you are joking or not, I sometimes do not know.
I gotta say, your English has improved a lot since we've been doing these extensive videos. You've gotten a lot of compliments, brother. So keep it up, man. There's no complaints coming from us.
Yeah, thank you. But it still remains my third. But it still remains my third language. Still remains my third language. Okay,
I can only speak one language, and I can barely speak that well. So
you do speak the DNA language, and that's a big thing.
Amen, brother. Amen.
Okay. Going to put because we're gonna make this comprehensive, so I have to explain again for the genetic students and everybody else. Okay, there's something that I forgot to explain. I think I'll explain it in less than 2 minutes, just quick, and then we'll go for the example. Standing for truth, you can give examples, or Neff, and I can give my examples. So you guys next after I explain it. Okay, again, the mutation rates. What are the mutation rates of DNA in there? Okay, the mutation rates of DNA is in the the most popular ones are in the mitochondria. Okay, they are organelles that are outside of the cell's nucleus. Okay, and don't forget, young Earth creationists, there is a design feature here that is seldom that you seldom hear. Okay, these mitochondria are outside of the nuclear DNA. Why? Because they produce free radicals, and we know what free radicals are. So this is a design feature that the mitochondria is outside of the nuclear DNA because
Good
they
point.
produce free radicals, and it will destroy the nuclear DNA if it's there. Okay? This is something you can use against evolutionists. They cannot refute this. I promise you that. They will
Actually,
say, they will
actually,
say,
Jeremy, yeah, that's a good point. Is that
they will,
kind of like humans will build their power plants away from like a school or something like that?
population centers. Population
Right.
centers.
Right. That's a design mechanism right there. How did the evolutionists explain that? How did that evolve? The mitochondrial DNA compartment being separate from the nuclear DNA compartment.
They would say they do not explain abiogenesis. That is, for them, that is called abiogenesis. They will say they do not explain abiogenesis. So that's where they lose. No, sorry. I said just less than 2 minutes to explain this thing. Okay, as we know that mtDNA or mitochondrial DNA is inherited from the creature's mother, right? Any creature. Us, yeah, any creature. And its mutation rates, okay, inherited, okay, and it mutates when per generation it mutates this mitochondrial DNA whenever it's passed from mother to offspring, okay, and its mutation rates can be measured to produce genetic molecular clocks. Remember rates, okay, rates. Clocks. Okay, clocks do produce rates. Okay, rates or correct me if I'm wrong, guys. With my English, rates and clocks are connected. So these mutation rates can almost accurately be measured to produce a molecular genetic clock by comparing rates in a few different animals. Okay, Dr. Nathaniel Jensen did demonstrate that a creation of not more than 10,000 years. 10,000 years is the max in his model. It's not 6,000 years. So I will not lie to anybody. It is not 6,000 years in Dr. Jensen's model. It is 10,000 years max. But of course, he believes in 6,000 years, right? He's honest, so he calculates it to 10,000 years, and we have to explain why it is 10,000 years, but
Well,
not 6,000.
Jeremy, real quick. A lot of your secular papers, like the Parsons paper, for example, they're not even young Earth creationists. They're just secular papers on mitochondrial DNA pedigree-based rates and studies, and they admit that the amount of mutations in the human genome, for example, only go back about 6,000 years. Even they say 6,000 years.
10,000 years based on Dr. Nathaniel Jensen.
No, like I'm looking at a paper right here, a Parsons paper. It's a secular paper, and
Yes.
it says 6,000 years.
Yeah, that was back in 1997. Yes, I've read that paper. But Dr. Jensen was, like I said, honest enough to say that his calculation is at max 10,000 years. Okay, if you forget standing for truth, Dr. Nathaniel Jensen did say that, and we have to narrow it down to 6,000 years somehow. Okay, so he is still recalculating. Okay, he is still recalculating so that it can go down to 6,000 years. I am trying to be honest to evolutionists here.
Well, the thing is, I don't know if he said that, Jeremy. Where did he say that? Because from what I've seen, I've even watched a few.
Replacing Darwin. He did say it in his book. Okay, it is somewhere. I forgot where, but I promise you, he did say that.
Well, because I'm looking at a lot of the technical papers here, and it's a moot point, but I'm not sure if I agree with it because I'm looking at multiple secular papers here, not just the Parsons paper, but subsequent papers that, based on the observed mutation rate, only go back 6,000 years. I don't see anything about 10,000. And then in these papers, they calibrate the date with the fossil record, and they invoke slower mutation rates. But even Dr. Robert Carter in Sanford would admit that so for example, the Y chromosome, Dr. Jensen's newest paper only puts the Y chromosome back to 4,500 years, which would be Y chromosome Noah. And the mitochondrial DNA differences, there's only a few. I think Carter's the one who has a paper out that goes or reveals the original Eve sequence, and those differences, the accumulating differences, are only 6,000 years worth. Is my point. I mean, I'm saying Dr. Jensen may have said that, but I don't remember ever.
I recall that. That is one of the most striking things that I read from his book. Okay, so I think I explained.
I think what he might have said is
you can ask him in the chats to confirm, or and then if I'm wrong, I know I'm right. I really remember this because he tested so many species, correct? So
okay. Wait a minute. Are we talking about human mitochondrial DNA rates or animals? Other organisms, because I'm just talking about humans. Okay, so maybe other organisms. Maybe he said there's a few that need to be recalibrated. Oh, okay, I thought you were okay. Yeah, I was just talking about humans. Yeah, humans, he would agree, go back 6,000. Maybe there's difference because, like you said, different generation times. Mutation rates are fairly consistent, but different generation times, different population history in different animal species. Yeah, those. The differences might be a little bit off that could be explained by other means. But the point is, yeah, so within humans and all of biological life, the molecular clocks only go back between 6,000 and 10,000 years, and not hundreds of thousands to millions of years like the evolutionists. So they got a major problem on their hands, that's for sure.
So thank you, Standing for Truth, my friend, for clarifying. So humans is 6,000, and the other samples also reveal 6,000, but some do reveal 10,000. Okay, that's what you said, right? Standing for Truth. Standing for Truth. Are you there?
I stepped away again for a minute, I guess.
Okay.
No, I'm just on mute, brother. Yeah, because you can have the floor, man. Whatever you got to say, go ahead.
Yeah, I think I'm gonna stop after I explain to the audience how we calculate mutation rates. Okay. Again, it's inherited only from the maternal line, the mother line of a creature, and yeah, I already explained. These mutation rates can actually be measured by uniformitarianism standards. I forgot to say that. Sorry. By today's rates. Okay, that is why this is more or less reliable. By today's rates, using uniformitarianism of evolutionists against evolutionists. So, okay, that is the strong point of this mutation rates that Dr. Nathaniel Jensen is saying. Okay, so you guys talk next, and we'll give. Okay, you guys talk, or you guys give examples. I'll give examples later, unless you want me to continue. It's up to you guys. You guys go. Please.
Well, I'll just say that in order to try to fix the problem, this is how severe the problem is, that scientists like Ota and Kimura did calculations to find out how many individuals a population would have to have, how many offspring, rather, an organism would have to have in order to solve the problem of the accruing degenerative mutations by increasing population so great that natural selection would then be able to purify the population and remove the degenerative mutations. And the number, you're not going to get this, individuals would have to, according to their calculations, would have to have 4 million offspring. just wait a minute. I'm sorry. Let me. Yeah, one of them came up with 4 million, but Alex Kondrashov says the parents would have to have a minimum of 44,000 offspring for natural selection to counter the occurring mutations, assuming only 2 of those 44,000 would be selected. Unbelievable. 2 out of 44,000. That's how wide you have to spread, how many offspring these creatures would have to have, and only 2 of them survive, be selected in order to combat the accruing effects of random mutation. I mean, what a fantasy world that is, right? Like, that's never going to be the truth, right? Ever. But this is, so the numbers really destroy the evolutionist. If you have an evolutionist tell you, well, because large populations are great. Oh, okay, so in a population, what? Mom and dad had 44,000 kids and only 2 of them survived? And that's how you fix it? Because that's the numbers. I'm sorry. That's what they come up with.
Isn't that funny how they just won't question? It's like that Grar paper I was talking about earlier. They won't question the time element, right? Because they're assuming that we've been evolving for millions and millions of years. So if the genome really is mostly functional and these deleterious mutations are accumulating, well, for natural selection to rid the population of all these deleterious mutations, you got to have thousands and thousands of babies per every woman's got to have all these babies. They're not questioning the actual theory. Like, guys, your theory is false. It's
right.
You know,
isn't that amazing that they don't question the theory? They can't do that. It's always there's some solution to it. Whatever it is, we'll find a solution, or no matter how bizarre the solution has to be, that's obviously what fixes it. Anything but question the theory, right?
Yeah, it's they can't question the basic assumption, right? And that's why they'll often say, well, how come young Earth creation scientists don't get published in these peer-reviewed journals? It's like these young Earth creation scientists, like Dr. Jensen, their model questions the entire theory and the deep time. Now, the ones that get published in these secular peer-reviewed journals, all these evolutionist buddies, yeah, maybe they're questioning little details here and there, but not questioning the entire theory. Nobody questions that. That's the holy grail to them.
Yeah,
it'd be like going to communist China or North Korea, and some anti-communist submits a paper into the peer-reviewed journal as to why communism is bad. Like, do you really think that paper is going to get published? Of course not.
Right. No, and of course they're going to deny the papers that are published, even by their own camp, in order to defend the evolution theory. So it's truly amazing that modern science has demonstrated, with this kind of information, accrued knowledge, that the mechanisms for evolution are nonsense. The problem that evolution has is that the idea that random mutations to DNA is what was the base mechanism for evolution was an idea that formed immediately after it was discovered what DNA was, and scientists realized that DNA is a package of information that prescribes the features of a living thing. That's when the evolutionists came up with the idea, oh, that's what it is. It's mutations to that package of information that create the new features. That's how evolution proceeds. But that idea was born long before long before the vast majority of scientific knowledge about genetics had been acquired. That idea came about in the 1930s and 40s. The mutation was the base mechanism of evolution. It wasn't until the 1960s and 70s that papers just started flooding the science journals describing the negative effects of random genetic mutation, and evolution then ran into a serious problem. How do we keep holding on to evolution theory when papers are hitting the journals left and right talking about the negative effects of mutation? So it was an idea, it's an antiquated idea idea that was formed before man really knew much about the effects of mutation. Now that modern science has advanced in that area, we see that the idea was never scientific to begin with, and it doesn't hold water.
It's like
Thank
you
you,
always. It's always like you say that the mutations, which they say is the creative force, is actually the destroyer. They say it's
It
the
is.
creator, it's the destroyer.
It is. Yeah. It's the
Okay.
actual opposite of evolution. It's devolving.
Thank you for adding data to our previous genetic entropy discussing, Left. You are good at this too, so we appreciate. Now, standing for truth, shall we continue with the last? We're going to be comprehensive, right? So, shall we finish the mutation rate? I already explained the basics for genetic students and everybody to understand. Do you want to go further? Give examples? I know you are very good at this also. So go, or you want me to start first? Up to you, my friend.
You can start it off, brother. You're on a good roll. You can start off.
Okay. As I recall, I will also give the evolutionary counter argument. Okay, that's what we did during our past four videos. We really covered everything: the evolutionary argument and our counter argument. Okay, no
Well,
matter how.
yeah, and yeah, you're right. And even in this one, I like that you've been bringing up the objections, right? Like bacteria, junk DNA, synergistic epistasis, all those rescue mechanisms. I do want to make a point. I see in the chat. I'm not sure. I kind of got a glimpse of it, but some people are suggesting that, and I guess evolutionists do suggest that, yeah, even though they're nearly neutral, they'll build up enough to the point where natural selection eventually sees it and then deals with it. Well, the problem is selection itself acts on phenotype and not genotype. It's either the whole organism or nothing at all. It's not just going to select one single nucleotide. It's phenotype. It's selection happens on the level of the whole organism. So the best way to put it is if you have a room full of people, okay? Now, even if natural selection were to just remove the most mutant people, you're still left with a room half full of people who still have roughly 60 to 100 more mutations than their parents, okay? And as I said earlier, if every single person is multiply mutant, and if every generation is then more mutant than the generation before it, selection is only going to be able to slow it down and do its best to halt the genetic degeneration. So yeah, eventually, even if selection does remove some deleterious mutations, these mutations have accumulated in the population as a whole to the point where there really is no stopping it. Things are going down and not going up. And that is what's fascinating is all of modern scientific evidence, what does it point to? It literally points to the decay and destruction of an originally perfect design and creation, just like our model with the creation of Adam and Eve with perfect genomes. If natural selection were to, because every single person on this planet is mutant. If natural selection were to just eliminate every single person on this planet, we'd go extinct. It can only act on the most mutant. And that's why any evolutionist who says, yeah, it'll eventually solve the problem, it's not going to, because you get rid of the most mutant, the other not so mutants, they're just going to keep accumulating. It's a downhill process. So, I mean, they can come up with any rescue devices they want. They ain't going to solve it. Like, these evolutionists just need to accept it. And I speak for Neff probably. We're probably even getting tired of explaining it. I mean, it's what we observe. And it's science denialism to say in any other way. So,
Yeah.
Go ahead. I just had to throw that in there.
What? You go first or I go first on some specific example? And by the way, before we give it, what, standing for truth, me or you?
No, you go ahead, brother. Yeah, I just wanted to
Okay.
address that argument.
All right, we'll give examples. The good thing about Dr. Nathaniel, I'll also give the evolutionary counter argument here. Okay, but their counter argument is not enough. Okay, now The good thing about there are two good things. There are two reliable. Uh, sorry for my English again. There are two reliable aspects. Okay, aspects of Dr. Nathaniel Jameson's calculation on mutation rates. Okay, the first one is he he calculates it based on several species. Okay, it's like he presents several evidence in court. Okay, so he's tested it on several species, and he's continually testing it, and things are panning out. Is panning out the correct English? Working out. Okay, because he has released some papers and fulfilled predictions. Okay, so that's the first good thing. He is using several examples, and these several examples are are more or less revealing the same mutation rate and the prediction that he is suggesting. Sorry, he is boldly claiming. Sorry again, not claiming. He is boldly attacking evolutionists because he said whatever. Forgot. Okay. Here, let's say from his book. Okay, roundworms. Roundworms. Okay, is one example. Okay, because okay, roundworms. Okay. All right. What was this? Roundworm. Roundworm. Okay. The base pair. Okay, we know base pair mutations. Okay, where he predicted from the roundworm mitochondrial DNA. Okay, it's like I said in my basic explanation, mitochondrial DNA, okay, mutation rate. Okay, he said that as I recall. Forgot what he said there. I'll just say it on my own words, exactly how I would say it. Okay. He said that I say that these roundworms underwent only about 9 generations per year, if I'm not mistaken. Consist these roundworms. Standing for truth, help me out. Because sometimes I forgot the exact data here. There are just too many numbers. Okay, if roundworms consistently went or underwent only certain generations, a lot of certain generations per year, then only around 45,000, yeah, 45 or 50,000 somewhere there. Sorry again, audience, I forgot the exact number. Okay, I have to check his book again. Then only around 45,000 mitochondrial DNA mutations would have occurred. Okay, over 18 million years. Okay, so 45,000 mitochondrial DNA mutations would have occurred over 18 million years. Okay, if evolution were true. Instead, okay, now if roundworms consistently went or underwent 100. Oh my goodness, the numbers. If I know it's somewhere between 100. Okay, sorry, audience. If those are roundworms consistently, what I mean consistently, uniformitarianly underwent 104 generations per year, then over 9 million mitochondrial DNA mutations would have occurred over 18 million years. Okay, see the inconsistency? Okay, now today, measured by today's rate, only less than 2,000 single base pair of mitochondrial DNA difference separate roundworm species. You see? Of course, evolution is gut rescuing device. Rescuing device which are, I have to be honest, which are somewhat reliable, or not really reliable, but because evolution is also cheap. They would they would they would uh, they would say that are you there standing for truth?
Yes, I was
Are
just
you
on
there?
mute, brother. Sorry.
I just want you to support me with this because we both are very familiar with this. So I really forgot the
So
numbers. So
what
you
you're
have to
saying
correct.
is the mitochondrial DNA differences that we see across species, humans, roundworms, for example, and many of the other examples that he's given. There's too few differences to be explained by deep time evolution.
Ah,
They've
yes, yes.
they've over predicted the amount of mitochondrial DNA differences or mutations. So they have to invoke exotic solutions like maybe natural selection managed to rid those species of all those mutations over time. Our question
Yeah.
would be sure, but for that to be science, what testable predictions can you make? Um, they will oftentimes invoke slower mutation rates in the past. But the bottom line is the fact that observed mitochondrial DNA mutation rates only take us back 6,000 years for humans, and then for some of the species, between 6 and 10,000 years. But there's always other factors at play too: generation times, population histories, extinction events, things like that. But point is, the bigger picture is mutation rate wise, molecular clock wise, in all of biological life is consistent with a biblical based model and an origin of just 6,000 years. Even the nuclear DNA, they've underpredicted it. So they're wrong by the nuclear DNA differences, and they're wrong on the mitochondrial DNA differences. They've overpredicted one, they've underpredicted the other. So there's all sorts of problems for them. Is that pretty much more or less what you're saying, Brother Jeremy?
Thank you very much for putting it in a nutshell because I explained it too technical. Thank you very much. I
No,
kind of lost.
you did good, man. I was following you the whole. Very good. You're doing.
Yeah. Do you want to give another example? Because I gave one. Maybe you want to give another. The flea species, because Dr. Nathaniel Jensen did. Sorry, is it flea or fly? FLEA. I don't know how to pronounce it in English very well. So
Fruit fly.
he gave several examples of it. Do you want to elaborate? But what you said, it was already in a nutshell. Okay,
Yeah, pretty much what I
sim-
said would
sim-
apply to all the.
simplifying
Yeah.
simplifying what I said. So thank you.
Yeah, no, I think you did good, and then I put it in a nutshell for the audience. Jason, good to have you, brother. Brother Neff, did you guys have? I think we've been pretty comprehensive on this topic. If you guys had something you wanted to say or any points you wanted to add, the floor is yours, guys.
No,
Well,
I'm
there's
just
a
here
lot
to
that can be
collect
said.
questions. If anybody wants to put up any questions, I'll collect them and save them.
Jason, do you want to ask any questions? Any questions?
No, mate. No, I'm not. I've just
I try to
I
answer.
haven't
Well,
been
there's
on here long, and I'm not.
Yeah,
And
me too.
I'm just focusing on collecting questions. So
How about standing first? Do you want to give the flea example? Because the FLEA sample that Dr. Nathaniel Jensen used, he has several examples of it in his book, as you know.
Let me interrupt just long enough to say that
Sure.
it's late and I'm beat. I'm gonna crash. You guys, good job and good luck to you. Keep doing what you're doing, and you guys, everybody have a great weekend, a great evening, and God bless you. Catch you guys next time.
Okay.
You as
Goodbye.
well.
yeah,
brother
God
Neff.
bless you, Neff. See you later, mate.
Always
Okay,
a pleasure,
brother.
man. We love you.
Okay.
We love
All
you.
right.
So get some rest, man. Keep up the
Thanks.
good fight.
Talk to you later.
Talk to you later, man. Talk to you.
We're almost
Computer
done
just died as
One-
usual.
So
One
standing
last.
for, do you want to give another example, or I give, or we don't give another example?
I think the bigger picture is important. I mean, to be honest with you, I'd have to pull up the exact numbers on the fruit flies. Like, I don't remember it off by heart, but if you know them off by heart, you can. But I think molecular clockwise, I mean, I think we've been pretty extensive enough. I did want to point out, unless you have something to say, I did want to point out genetic entropy in the fossil record and how that explains the so-called pre-humans that evolutionists look to. But before I get to that, Jeremy, you can, whatever you got to say, brother. If you had another point you wanted to make, or the floor is yours.
I have to be fair to evolutionists and let young Earth creationists know the objection to Dr. Nathaniel Jensen's data. Or I could give another example from the flea species. I recall two out of several of Dr. Nathaniel Jensen's flea species examples. Do you want me to go that route? Last example, and then I'll give the evolutionary objection. So, or are we going to end it now?
Are
It's
you
up to you.
going to? Yeah.
No,
If you,
go
here's my
for it.
recommendation: if you give their objection, answer the objection afterwards.
Yes. The
Okay.
answer to their objection is actually. So I'm not going to give any more examples. It's done.
Okay.
Okay. The round-worn example is enough for now, and that is the technical explanation. And standing for truth gave you the simpler explanation. All right, so it's perfect. Okay, we could give more examples next time.
Well,
Okay,
and
now,
you said you
sorry.
wanted to give the Did you say you wanted to give the evolutionist objection to it?
Sorry, objections to.
Oh.
Sorry.
Oh, I thought you were going to give the evolutionist objection to what we were saying. Maybe I was wrong. Sorry. I thought that's
Yeah,
what you.
I wanna give. We want to be this video to be comprehensive, and we were already comprehensive about genetic entropy above. And Nef added a few things, and then the evolutionary objection to genetic entropy. I think we have already covered those, and and then now the mutation rates. I gave a technical explanation, one example, and I gave the basic explanation on how we do these mutation rates, how Dr. Nathaniel Jensen do these mutation rates. I gave one example of his example in his book on roundworms, and then you made it easier to understand, in which I forgot to put it in a nutshell. So I think we're also more or less complete, unless you want to add something. And before you add it up, I'm gonna go for the evolutionary argument against Dr. Nathaniel Jensen's argument. To be fair to evolutionists and for young Earth creationists to not be shocked.
Yeah, no, I think that's a great idea, brother Jeremy. If you wanted to tackle that, you've done a great job, Jeremy. I appreciate it. As always, man. You're always you're an asset to this fight. So God bless, brother.
Yeah. Okay.
I just added in a link to the synergistic epistasis to a write-up of what Dr. Sanford says about synergistic epistasis. Did you cover that?
We did,
Yes,
yeah. Brother Jason, thanks so much. Yeah,
I did give a very short nutshell about that.
And then, well, we both touched on it for probably a good 15, 20 minutes. So that's good, Jason. Yeah, their main arguments, which we covered, is the junk DNA, the synergistic epistasis. There are other artificial mechanisms like mutation count mechanism. For example, bacteria and mice. They'll look to, why are bacteria still here if genetic entropy is true? So we've pretty well covered all of their major objections.
What were they saying? Were they saying basically what they're saying is, and I heard this said before, and this is probably off the subject, is that you've got the germ or the viruses in our DNA, the ERVs, but the ERVs across all mammals. And without the ERVs, as far as I know, that all mammals without the ERVs cannot develop the placenta. So that means that there's no babies. So ERVs being, like what they say is because ERVs are across all species, that proves evolution. Well, then how come we need ERVs if they only came in, how many millions of years ago was it that ERVs only came in, about however many millions or 500,000 years ago or something like that. So for millions of years beforehand, the placenta couldn't develop properly, so therefore no babies.
Yeah, exactly. They weren't having any babies for millions of years until the ERV infiltrated the genome, and then a function was co-opted. So, Good job, evolution. Yeah, no, ERVs regulate gene expression in a number of things. In the gastrointestinal tract, ERVs are important in the placenta, like you said, in the testes, the mammary glands. So no, these ERVs are making very significant contributions to normal physiology, because if they're playing important roles in development, well, you know, that certainly suggests design. Therefore, if they look to ERV shared nested patterns, well, it only makes sense. If these are functional DNA elements, we're going to expect chimps and humans to share more ERVs than chimps, humans, than humans and dogs, for example. So no, that's a great point. That's a great point, brother Jason. So yeah, Jeremy, what was the next thing you wanted to talk about?
The evolutionary, the most popular evolutionary argument used by Herman Mace and Dr. Dennis Venema on the Dr. Jensen's calculations is none other than the substitution rate. Dr. Jensen, they say, did not put the substitution rate. That is the number one argument. There are more, but as Dr. Jensen said, if evolutionists can use uniformitarianism, why can't we? And Dr. Jensen's uniformitarianism calculations on these on the insects in his books are all saying the same thing. So no matter what evolutionists say, if these numbers do correlate with each other, then you have no objection, especially when you use the substitution rate. Now, what is a substitution rate in the evolutionary literature, or in general? Substitution rates are they are like fixed mutations, or what's the English for them? Fixed synonymous mutations. Okay, it means that the DNA is having the same or almost the same meaning as to another word or phrase in the same language. Okay, yeah, so they give synonymous substitution. They say that these synonymous substitution would, because Dr. Jensen presented graphs in his book, so it would reduce those graphs, because the graphs in Dr. Nathaniel Jensen's book on the roundworm, that I wish I could show the graph, are really huge. I gave the numbers. If people were listening, the numbers are really huge. It really contradicts the evolutionary mutation rate. Okay, so it's a synonymous. So their counter argument is synonymous. Sorry, the English again is what's that again? Synonymous substitution. To be specific, okay, it is substitution rate is synonymous substitution. They are changes to the DNA sequence of a gene that do not change the protein produced by that gene. Okay, they are usually used to estimate that mutation rate. So evolutionists use substitution rate. So they want Dr. Jensen to also use substitution rate, because the evolutionists use substitution rate. Okay, there are reasons why Dr. Jensen did not use substitution rates. Okay, and yes, he did omit data like substitution rate, but it is reasonable to omit substitution rate because substitution rate is not consistent. It is not even consistent with uniformitarianism standards. It gives different rates. You know about this standing for truth?
Yes. Well, my question for you, does the evolutionist calibrate the number of substitution rates with the fossil record, with their phylogeny assumptions of when they believe humans evolved? Like when they believe there might.
Can you re-ask the question? What do you calibrate the substitution rate? Yeah, they do. They do apply it to everything, actually. And they do get really different rates. Is that the answer is that it? Is that
Yeah,
the question?
yeah. That is because with their way of explaining the data. So, for example, Dr. Jensen, the point in question is the time frame, is the geological column. That's the point in question. So we don't assume deep time. We're just looking at the observed mutation rate, and we are we're calculating it back to see if it fits with deep time or with biblical time. So the fact that the observed rates go back to biblical time, we have no reason to adjust it or calibrate it to that very point in question. That would be circular reasoning, So the evolutionists, they'll look to the observed mutation rate, and then they'll admit oftentimes, sure, it goes back a few thousand years, 6,000 to 10,000 years, but we know based on the fossil record, in phylogenetic systematics, they'll say that humans evolved 250,000 years ago, or for example. So now they calibrate the data with the time frame, which is something we don't do because we don't assume the time frame. So why would Dr. Jensen do that? Does that make sense, Jeremy? I'm trying to explain it the best way I can, but
yeah, I think so. And they are assuming today's substitution rate. We do not know the substitution rate in the past.
right.
Okay.
Yeah,
So,
it's all imagination. It's just all
yes.
pure imagination.
Well, that's what I mean. Doesn't the observed substitution rate? They want us to assume their model and therefore assume unrealistic substitution rates in the past. But we can't. We're just assuming, for the most part, consistent mutation rates. We're assuming uniformitarianism for the most part when it comes to the molecular clocks.
Yes.
Yeah. The key to the past is what's happening in the present. That's what evolutionists say.
Right.
Yes. It's not only the substitution rate. There are a lot of other examples in the fossil record, even in astronomy. Everything. I've studied these things, and I have seen them really cherry pick data, which like the substitution rate that they intentionally use this against Dr. Nathaniel Jensen, okay, without knowing the substitution rate of the past, okay.
Now, does the substitution rate of the present, is that consistent with the 6,000 years for Eve?
We have not. I have no data about that. I'm honest. I have no data about that.
We don't know.
We don't. I don't know. I don't. Maybe others know, but I don't know.
Okay.
Sorry.
No problem.
Sorry.
I thought that I read a study in Matt's book that showed that they got 6,000 years back to Eve, but that's not what you're talking about, is it?
That was the Parsons, I think, that Standing for Truth was referring to, if I'm not mistaken.
Oh, sorry, I got it mixed up. Sorry.
No, yeah, Jason, you're right. Yeah, but like pedigree-based study, like just looking at the mutation rate, how many mitochondrial DNA mutations are passed on from generation to generation. They can look at multi-generational studies, grandmother, mother, daughter, see how many differences arise each generation. Take that back to 6,000 years. Jeremy is saying that Dr. Jensen, in his mutation rates, and the mutation rates he derived from the secular papers, he says Dr. Jensen ignores substitution rates, and he needs to account for those. And if he did, then apparently that would confirm deep time evolution. But Jeremy, you're pointing out that we don't know.
We cannot incorporate substitution rates because we don't know the substitution rates of the past.
Exactly.
So what we're doing is more scientific. We're not just going, oh, well, we'll just make up whatever fits the biblical timeline and to hell with everything else, which is what the evolutionists do. We'll just make up whatever fits the evolutionary timeline, and all the little drones out there will just swallow it down.
Right.
My goodness, I could give a lot of genetic, geologic, and astronomy examples about these cherry-picked data. Okay, so again, substitution rates, which they call synonymous substitution rates, which are DNA. Oh, I already explained it, so I'll just proceed. They are changes to the sequence of the gene that does not change the protein that are produced by a gene. Okay, so they are evolution is used to estimate that mutation rate, even though it is because 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 other rates that they use give a larger discrepancy. So this is the closest they can get, and the closest they can get is not even close. Okay, so so but this is a trade secret. Okay, now
So we use real science, they use fake science, basically.
Yeah, and the thing is, as I pointed out, you know, we are using real science, observable science, empirical science, the empirical method when it comes to mutation rates. So the fact that the mutation rates that are observed in the mitochondrial DNA, in the Y chromosome DNA, is consistent with the biblical time frame, that's it, case closed, because we don't have any need to now calibrate it with their deep time assumption. Doesn't make any sense. The fact that they want to assume deep time, take the observable mutation rate and say, okay, it goes back 6,000 years roughly. Okay, but we know evolution in deep time is true. Now we got to calibrate it with unobserved substitution rates, unobserved mutation rates, unrealistic mutation rates to fit with our model of deep time evolution. That's a
and they call that science.
Right, yeah. Yeah,
and they call that rock solid science, and then they laugh at us and call us morons and idiots and creatards when we don't believe in that stuff. Well, we say, well, that's not science. That's imagination. You've made that up to fit your narrative. I mean, that's not science. That's theoretical science at best. I mean, you know, not even theoretical. It's pure imagination, because theory needs something to back it up at least, and there's nothing to back that up at all.
It's antagonizing, isn't it? It's annoying that they
antagonizing is not the word.
I mean, epigenetics and those environmental factors can change this substitution rate many times in history. Environmental and those epigenetics can change the substitution rate so many times in world history. So who are they to say that this is omitting data? Okay, so they say that. So they often use this to estimate the mutation rate, because the other way to estimate mutation rate are reveal more discrepancy. So this is the best one, the closest one. It's not even close. They often use this. Okay, despite the fact that some of these mutations, synonymous mutations have fitness effects. Okay, so I think I'm done.
Because well, really, their only explanation can be one of two things. How many? They'll have to say in the past natural selection was more efficient. So there were more mitochondrial DNA differences, but natural selection was working better, and it got rid of all these DNA differences, and that's why we have so few. Or maybe a combination of both. Or they can say that mutation rates were 10 to 20 times slower in the past than they are now. So both are appealing to the unobservable past. So it's post hoc, ad hoc, it's a rescue device. But the best thing to ask the evolutionists when they invoke those mechanisms is, what testable predictions can you make? What testable predictions can flow from those explanations? when does your mutation rate, when does your clock slow down? When does it speed up? You know, how many mutations, for example, does natural selection have to eliminate during your deep time evolutionary time frame? You know, make a prediction. Let's be able to test it, because guess what? Dr. Jensen's so confident with his model that he's pointed at African people groups like the Khoisan peoples, and he says, I predict that their mutation rate and their DNA will change this much in this generation, for example. Or he'll say, point me to an animal in the wild, point me to a fox, you know, give me its generation time, for example, and I'll tell you exactly how fast that fox, that specific species mutates or changes. You know, he's taking it to them. He's the one making the predictions. Dr. Vanima in the debate, he even admitted, he said, you know, you're gutsy for making that prediction. And Vanima admitted he can't make that prediction because Vanima said that there's too many factors at play, too many things to consider. Okay, so be it. The fact you're not making predictions makes your explanation non-science. While Dr. Jensen, his explanation is science. Because Jeremy, as you always brilliantly say, the gold standard of science is testable, falsifiable predictions that are repeatable as well. And yeah, no, that's all I got to say there.
Yeah, I have nothing left to say unless Jason, do you have any question? Sir Jason? Is he still there?
He may have moved from the computer. So we are going on 3 hours, but yeah, you're right. I want to be as comprehensive as possible, and I got to be able to get a little bit of sleep tonight. So the last thing I would say, and then Jeremy, you give me your thoughts on this too, is the evolutionists will oftentimes look to pre-humans, and they'll look to pre-humans like your Neanderthals, your Homo floresiensis, the hobbits, for example, Homo naledi, Homo erectus, Homo heidelbergensis, whatever, you name it. And then they'll look at these so-called primitive features that are seen and in these so-called pre-humans and say that, you know, they are our evolutionary ancestors, for example, when in fact, the very thing we've been talking about, genetic entropy, best explains these as not being pre-humans, but actually humans. They were members of our own species, 100% human, but they were undergoing accelerated genetic degeneration due to severe and extended inbreeding. And it makes sense because post-flood and post-Babel, okay, you get these populations of these individuals that they migrate after Babel and they become isolated, unfortunately. And therefore what happens is what's called founder effects, as well as accelerated genetic degeneration. Because when you're inbreeding, then many recessive mutations are now coming to the forefront. And founder effects, for example, guarantees the descendants will display a variety of genetic abnormalities and defects of the founding individual. Now, this is exactly what would be expected given the post-Babel dispersion, which, as I said, would include inbreeding for many generations. And therefore, that genetic degeneration due to the inbreeding would give rise to these anomalous populations that the evolutionists point to and say, hey, look at this, you know, this is our evolutionary ancestor. No, it's modern humans that unfortunately have experienced this type of reductive evolution or genetic entropy. So that best explains your hobbits, your naledi, your erectus, your Neanderthals, because all of those human, so they look at it opposite. They look at it like, okay, Homo erectus evolved into all your different pre-human people groups, when in fact, no, humans, modern humans, well, I don't want to say devolved because they are modern humans, but changed based on these genetic related effects. And it's not fun, you know, but this is just what happens given the specific situations associated with these human populations that unfortunately experienced founder effects, isolation, and inbreeding. And honestly, any feature that they can point to, for example, the reduced brain sizes, right? They'll line up all the different so-called hominids and show smallest brain to biggest brain in some evolutionary history, when in fact, in these types of situations post-Babel, these isolation and inbreeding related situations, reduced brain sizes are actually beneficial in these types of conditions, because brains, as you guys know, they're energetically costly. So all of these features they point to, this is evolution in reverse. And any one of these anomalous features, these pathologies, these variants that they'll point to, can occur easily in isolated populations. And as I said 3 or 4 times, this is exactly what we expect and predict in the biblical model post-Babel and post-flood migration. It's actually fascinating that this is exactly what we find. So this data, point is, human evolution has been absolutely demolished, since it's just more evidence of genetic entropy and genetic degeneration. Because remember, inbreeding is like a sneak preview into where we are going genetically as a species, okay? Because inbreeding reveals the hidden reservoir of genetic mistakes. So you will experience, inbred
because the alleles line up more efficiently, and
Exactly.
and it brings it out. Yeah.
Brings it out. So you're going to experience genetic entropy quicker. That's why there's these examples of like butterfly populations that are isolated, mammoth populations that are isolated, and these unfortunate people groups, modern people groups that became isolated as well post-Babel. And this is exactly what we see. So just like the hobbits they point to, those Homo floresiensis that were like 3 or 4 feet tall, that island on Flores, I think it is, they experience what's called island dwarfism. And even on that island, I believe, I'd have to look into this one just to double check, but I'm pretty sure that there's elephant populations on that island that are also that have also experienced a reductive evolution. They're dwarf elephants. So if that doesn't corroborate with the data, right?
Hey, what's this mean? Substitution load is selective, elimination tolerable by the population size. Somebody said that. I don't even understand what that means.
I explained that already.
Oh, okay. All right. Okay. Forget it. You've already explained it.
Said that. Is it an evolutionist who said that?
Christopher Pock.
I cannot see that actually.
Christopher Pachari, or Picari, I don't know how to pronounce it, said that.
There's nothing to contradict what I said. And standing for truth, everything you said is getting closer to the haplogroups.
Right. Yeah. That would go to the haplogroups with L0, L1, L2, L3, L0 being Eve. You can see that, like your Neanderthal populations or Denisovids, for example, they branch off of those L nodes because and they have
no. Yes. Sorry. Yes. When they you forgot to say this. Those inbreeding, actually, you forgot to say where they inbred. This inbreeding happened. It's in Africa.
Right.
It's in Africa. So,
well, it could have happened in wherever these different populations, because we already know where a lot of them are found. Like the hobbits, for example, Homo floresiensis. Neanderthals are found in other places other than Africa, but the African line, they look to the L nodes, and those lines are longer, right? And they say that's all Africans because the higher genetic diversity, for example. And I think your Neanderthal, when you look at the DNA, it branches off of that even more. So they were even more genetically diverse. But either Neanderthals experience accelerated genetic degeneration due to inbreeding, which makes complete sense, or the DNA is not trustworthy, as Jensen points out, because even DNA stored today starts doing funny things after a year. degrades? Is it trustworthy, kind of thing? But you know what? Even if it is trustworthy, it still makes sense because of this hypothesis. And it's funny because I always bring.
Well,
Go ahead.
as DNA dies, it wraps around the histone and it becomes really tight and it binds up and it snaps in different places. And those and they've got all the DNA inside the cell, but it's all snapped and broken because it's bound up. And the way that they get it back together is they'll get the human genome framework on a computer, and they'll take those little pieces and go, oh, well, that piece matches up here and that piece matches up there, and that's how they piece back together, say, Neanderthal DNA or whatever, because they know they're human. That's basically how they do it. The DNA is still there, but because after death it binds up real tight and just starts snapping, so it's all sort of broken up in a jigsaw puzzle, and you need a framework to put it back together. That's why if they find dinosaur DNA, what framework are they going to add it into to work out where it goes?
Right. Good point. Good point. Did you have anything to say to that, Brother Jeremy? Yeah, my whole point of that.
No, I was just letting people know about ancient DNA when you were saying about the problems with it. Like, they do find ancient DNA and they have pieced together Neanderthal DNA, and that's how they know that they're very human, because they basically just matches up with our DNA almost perfectly. So
it falls right into the human variation. Yeah, I'm definitely one. I'm one who falls into the camp that I believe that for the most part we can gather some honest data from it, some accurate data, and that's why I believe the data gathered from the DNA evidence, the ancient DNA, suggests that this is indeed the case, that many modern people groups migrated after Babel, became isolated in different parts of the earth, and unfortunately that results in founder effects, inbreeding, and accelerated genetic degeneration, and would ultimately explain all of the so-called primitive features that evolutionists point to as being primitive, when in fact they're not primitive. These are not
primitive at all.
Right,
They're
exactly.
just ancient human beings. I mean, it's funny how Denisovan children, Neanderthal children don't have brow ridges, and one dental or a dentist, scientist of dental surgery, he said that he believes there's evidence that they didn't even mature until they were in their 20s and 30s.
Yeah,
like we're maturing. Some kids are maturing today at 10. Having their periods at 10. He doesn't believe that they matured later, they were stronger, faster, they lived longer. So if you've got people maturing in their 30s and brow ridges don't grow they grow as you get older, because Denisovan children don't have the brow ridges, well that would suggest long ages, that they lived long ages, and so on and so forth, with healing teeth, and you can't, whatever. Sorry.
I think that's Jack Cuozzo. I was even reading that book last week, Buried Alive. Great book. Love that book. I think he even points out the teeth enamel regenerates. Because I think the evolutionists will oftentimes point to the teeth enamel.
Yes.
But
yes, the teeth enamel, there's a gene in us still today that's now broken, but there's a teeth enamel repair mechanism. So yeah, they can't date their age they notice how they always say that most Neanderthals they find died at 35 or 40. Well, if your teeth are healing,
exactly,
then how are you going to date, a 900-year-old person if his teeth are healing when they crack and like a muscle heals?
Yeah, they're taking what teeth enamel does today and using that to come to their conclusion. When in fact, if what is true as is suggested, I think it's Dr. Jack Cuozzo, he's the dentist, so he'd be the guy to look to as an authority on that. Yeah, if it regenerated, then they're coming to an inaccurate conclusion. So, yeah, good point.
I'll post the link to the video in the chat.
Did you have any points to make, Brother Jeremy? The floor is yours, my man.
You guys are not sleeping yet?
No, I think for me, I've said everything I need to say. I think we've been going 3 hours and 8 minutes. Wow, yeah, I got to get some sleep. But unless you guys had anything, I think Matt suggested that he was maybe, because I'm 3 hours. Like for me, it's 3 a.m. But for Matt,
Oh yeah.
I think
It's
for
midnight.
Matt, it's only 12 or it's only midnight. So he suggested he may start a new stream on his channel if you guys wanted to join.
Yeah, I'll
Just do
join
this
over
all
there.
night.
So.
It is only 7
Go to
p.m.
bed,
here.
man. Come on, we're just gonna keep you up. I've got so much stuff I could keep you awake for the next 10 hours. So you may as well just go now. So.
Oh, it's been rough the last couple months. I've had some 6 a.m. nights, some 7 a.m. nights. Because you're right, when we get in here, we can go on, and 3 hours is almost nothing anymore when we get
it's a drop in
talking.
the bucket.
All right.
Just don't get overtired, because once you get used to that night, sleeping at night and being awake during the day, oh man, it's hard to break out of. But it depends on what sort of job you're doing. If you're doing a job where you work night shift, it'll work out fine for you. Anyway, man, good night. Love you, brother. And then thanks, Jeremy. You've been really awesome, and I enjoy listening to you guys talk. I learn a lot.
Well, I learned a lot from you too, Jason, and you as well, Jeremy. I mean, you guys know your stuff, so I think that's why it's always a good time and extremely informative when we all get together. So thank you.
I only know what you and Jeremy and Matt have taught me, so it's not really. Yeah. Anyway, I'll see you later. I'll pop out now and try to talk Matt into getting another stream going. No, you don't want to. You want us to go to bed
Standing
early.
question, can we speak in private for a while?
Sure.
Okay.
Yeah.
All right. See you, man. See you. See you, Jeremy.
Maybe later. I'll see you.
Brother Matt, if you want to shut it down. And I want to thank the audience for sticking around. Any questions, as always, let us know. We do our best to answer all the questions. And I hope everybody enjoyed and got something out of this. So good night and God bless to our faithful listeners. We appreciate you. And Jeremy, thanks for joining as well, brother. It's always a pleasure.
Yeah, we have to go offline. I mean,